morning. Can we stand for the national anthem?
Uilinde Jumuiya Afrika Mashariki, tuwezeshe kuishi kwa amani, tutimize na malengo yetu. Jumuiya yetu sote tuilinde, tuajibike, tuimarike, umoja wetu ni nguzo yetu, idumu jumuiya yetu.
Thank you. You can be seated. We are going to have a prayer from one of our students, Naomi.
Let us humble ourselves and pray. Father, in the name of Jesus, we thank you, Lord, for this day. Thank you for each one of us. Lord, we present ourselves unto you, that you will help us to deliberate our purpose in this conference for the glory and honor of your name. Receive glory and honor even as we begin. Walk with us today, walk with us through to tomorrow. We thank you, we honor you, for it is in Jesus' name we pray and believe. Amen.
Thank you, Naomi. Vice Chancellor, Deputy Vice Chancellors present, our guests, directors, deans, heads of departments, members of staff, students, presenters from outside the university. Welcome to our 9th International Interdisciplinary Conference today. Our Vice Chancellor, this is our second conference this year. We had one in November that involved 3 schools: the School of Arts and Human Development, the School of Business and Economics, and School of Education. So this conference today actually is about 4 schools, but we've also brought in the other 3 schools. So today, as you can see from our theme, it's about digitalization. We want to welcome you to preside over this conference, and there are many committees that have come together to bring this day or this conference to fruition. We have our conference committee headed by Dr. Mulindi, if you can stand. And members of the committee, kindly just stand and wave. We also have our school research chairs. Ones who are present can just stand up and wave. Then we have the school, the Board of Research and Innovation that is chaired by Professor Gelas Simiu. So those are the committees that have come together to bring this day to fruition. So we want to welcome you, and I would like to welcome the chair of the Board of Research and Innovation, to invite the Deputy Vice Chancellor, Planning, Research and Extension,
the Vice Chancellor, the Deputy Vice Chancellors present, the invited guests, presenters, colleagues, good morning. This is a great pleasure, and we are greatly honored to host this conference, and we are happy that you are here to grace it. Without wasting much time, I just wish to appreciate everyone who has made this conference preparation a success. And at this moment, I would like to invite the Deputy Vice Chancellor, Planning, Research, and Extension, Professor Philip Rapuru, to take over from here. Thank you very much.
Good morning, ladies and gentlemen. Good morning. Yes, sometimes I go to church. If you don't see the hands up, then people are not hearing. If you are leaving, please just wave it like that. Thank you very much. We are very excited to have this conference going on, and also to have the blessings of our Vice Chancellor, who has a lot of passion for research. And since I have him here, I think I should not speak anything much, because I know we are a bit late but we are going to try and see how we can catch up with time. So welcome so much, ladies and gentlemen, and allow me not to speak anything right now, and to call the Deputy Vice Chancellor, Academic and Student Affairs, just to say something small and welcome the Vice Chancellor. Thank you. DVC, please.
Good morning. The Vice Chancellor, University of Eldoret, Deputy Vice Chancellors in our midst, Deans, Directors, our invited guests. I would like to welcome you for this academic exercise. As we know, research is an important component of our scholarly lives, and I want to appreciate that we have a chance to interact and share our research findings and ideas as a people who want to move forward. My task this morning is to welcome the Vice Chancellor, Professor Cheruiyot. Please welcome to address this session and formally open. Karibu sana.
Thank you very much. Thank you, DVC Academic and Student Affairs, organizing committee of the conference, Deputy Vice Chancellors present, our keynote speakers from Calro and also Kenyatta University, all the delegates, researchers, presenters, our postgraduate students, ladies and gentlemen, good morning. It's my great pleasure, and I'm delighted to be with you this morning. As you're aware that University of Eldoret prides itself as a hub of innovation, research, and academic distinction. And therefore, we normally have two research conferences, international research conferences annually. And I want to thank the team led by the Director of Research and Innovation for organizing this conference. And of course, as you are aware, research is a core mandate in our university, as it is in many other universities worldwide. And this conference, the 9th International Interdisciplinary Conference, whose theme is Innovation for a Sustainable Future, unlocking scientific technologies in science, agriculture, environment, and engineering is a testament of our commitment towards advancing the needed innovations for a sustainable future. As researchers, as academicians, as industry, we are called upon to come up with solutions to problems that afflict our generation and our society. As you may all know, universities and research institutions are centers for innovations and hubs of intellectual advancement in various disciplines, and therefore we are called upon to be able to come up with solutions to nagging issues and challenges in our contemporary society, such as climate change and many other issues. And I know that many of our presentations, which I've seen, are very, very interesting, and they are actually they are a source of solutions to a lot of challenges that we face. We have seen a lot of them actually original, laced with creativity, and therefore they're going to be very informative for all of us. Through this conference, we are embarking on a voyage of discovery, uncovering remarkable initiatives and pioneering endeavors that will shape the future of our immediate surroundings and beyond. From our own researchers from all over the world that are here, we are very cognizant of the fact that we need to move beyond research, and we need to move to innovation and commercialization, so that we move to the third generation universities that we want to be. Because if we just do research and there is no innovation and commercialization of what we are doing, then we are wasting a lot of resources, because knowledge for knowledge's sake is not enough. However, therefore, I would like to welcome all of us, and I would like to encourage that we participate, we share, we discuss, we deliberate, we share our experiences, we actually extend the knowledge and ensure that when we leave, we are living, better than we came. And I would like to also welcome you to visit our institution. We have a lot of innovations that in your own time, those of you who are not from here, you can always, been shown a lot of innovative research that is going on. And maybe I would like to invite the Director of Research to organize that. And I think going forward, we should be able to publish also, the presentations that we make. I'm sure there is an avenue for publication. And if we do that, then of course that can have higher impact. What we are interested in is to have high impact research and publications that will shape even the discussion, that will shape the future of our countries and our universities. And therefore, I would like to take this opportunity to wish each one of us very, very fruitful discussion and very fruitful presentations, and a fruitful time here for the next 2 days. And I would like to take this opportunity to declare this conference officially opened. Thank you, and may God bless you. Thank you very much.
Can we please give another round of applause to the Vice Chancellor? Thank you so much, our dear VC, for setting the stage for us to start our conference this morning. Ladies and gentlemen, allow me just to introduce the keynote speaker. That is Dr. Richard Oduor. I was trying to read what I'm supposed to read, that there's a big gap, but I discovered why the gap is there. This guy went to primary school and passed, but never went to a secondary school, and was scrambling notes from the village, from the students who are coming from schools, and read, and read, and read, and passed, and went to the university direct. So if you don't see anything about his earlier education, don't worry. But I now know why, what happened. But he started now from the university. So allow me to read what is here, that Dr. Richard Oduor has an extensive experience in the field of biotechnology. His active research focuses on using modern biotechnologies, including genetic engineering, to confer innate tolerance to local staple food crops against environmental challenges such as diseases, drought, aflatoxin, and striga. He holds a patent on a stress-inducible promoter with the World Intellectual Property Organization and chairs the Kenya University Biotechnology Consortium, a professional body that brings together experts in biotechnology from public and private universities to promote safe use and application of modern biotechnology in Kenya. Dr. Richard has served in several boards. Currently, he sits in the board of Global Biotechnology Transfer Foundation based in the UK. He is also a board member of the Kenya National Bureau of Statistics and chairs the Finance and Administration Committee of the board. In the year 2017, he was among the 52 African scientists celebrated by the South African Department of Science and Technology for their outstanding contributions in advancing research in the entire continent of Africa. Richard has won several research grants, supervised several graduate students, and published widely in the discipline. Apart from being an associate professor of molecular biology in the Department of Biochemistry, Microbiology, and Biotechnology, Richard previously served as the Director, Research Support and Dissemination before his current service as the Registrar, Research, Innovation, and Product Development at Kenyatta University. Richard holds a PhD in molecular and cell biology from the University of Cape Town, an MSc in biotechnology, a BSc in biochemistry from Kenyatta University, Kenya. In addition, he has a postdoctoral training in drugs discovery from Pfizer in the UK. He also holds an executive Master of Science in Management and Organizational Development from the United States International University, Africa, a graduate diploma in Forensic Sciences from Blackford Institute, UK, and a certificate in the Science Communication from the University of Cape Town. He has served as a honorary visiting fellow at the University of Leicester in the UK. Ladies and gentlemen, let's put our hands together and welcome Dr. Richard Oduor to present his lecture.
The Vice Chancellor, the Deputy Vice Chancellor is present, the directors, professors, guest speaker, Paul, colleagues, ladies and gentlemen, good morning. when the DVC was introducing me, for a moment I felt like that's too much. I think if I knew it was going to be read, could have written it differently, and given it just a quarter of a paragraph, and that's just it. We are here to engage actively, and I really want to thank the organizers, and thank you, Lizzie, and allow me to use the first name just to display the fact that this relationship works and has worked for several years. Thank you for always being here, and mentorship and all that. Yes, I am Richard Oduor. Like you have already heard, I work for Kenyatta University. And the topic that I have for today, why is there not? Is there something about the slides that is not showing the right? Let me see whether the second one does so that it becomes, that's fine. So if that's the case, then it's easy. We have a gentleman here called Paul Mireji. He will touch this very quickly, and it will work. I think based on, you are right, it's not your screen. It is this one. Right, so I was invited for all those things you've had. The only thing that has brought me here is that certificate in science communication. All those other things are now made irrelevant because it's about communicating research for food security. And what is important, therefore, I don't know, the Vice Chancellor, am I blocking you? Are you able to? You're fine, thanks. Anytime we talk about two things that are critical for me here is communication and also at the same time food security. So my background, like you've had, it's about genetic engineering. And the reason why this is important is because it's an ongoing discussion in this country. Anytime you mention GMO, people start developing things. And today, by the end of this discussion, if you still have not gotten this, then during lunch, we can compare notes so that you understand that it has never been this difficult, the conversation around genetic engineering and molecular biology. What is it when we talk about food security? For us to say that we are secure in terms of food, four things are critical. That food must be available in the right quantities and quality. It must be accessed. Accessibility means that you have the income enough to buy if you don't produce. At the same time, that food must be utilized. And utilization here means that you now convert it into other things. You can process it into finished products and all that. That is utilization. And the last bit is stability. And stability is very interesting, colleagues. Stability means that how stable are you as a country that a small little crisis does not throw you off balance. For instance, right now we are having flood. And just because of that, the country holistically is actually now worried that we are going to see a lot of famine because of just the flood. It's just a single event. That means, therefore, that we are not stable. Just by small little confusion around the quality of and I'm here saying that in quotes, confusion about the quality of the fertilizer, the entire country is panicking that we won't get the right harvest. Those are single events that shouldn't disturb the food security of a country. And that therefore means we cannot claim and say we are food secure if a single event is knocking us off. So as you talk about food security, it is important for you to also start thinking about food safety and quality for that matter. You can have a lot of food, but it's not safe. Again, that is important because the food availability, food access and utilization, and the processing bit of it from the production to utilization comes in with the quality bit of things. And therefore, what are the barriers to food security and food safety? This is where the discussion starts. And that is where now we come in as molecular biologists. The barriers are very interesting. If somebody has not understood that the climate is changing, then you need better goggles. Why? Because we are undergoing floods right now, and it is ravaging this country, which is so sad. What this means, therefore, is that the areas that were cold are getting colder, and the areas that were hot are getting sizzling hot. That has an impact in anything that has DNA. It means that crops that were previously very tolerant to certain diseases and pests are now becoming susceptible, and pathogens that were not actually, also very susceptible are becoming tolerant because they have to conform. Later on, you might understand there is a lot of what we call epigenetics, where how is the DNA and the genome behaving in the face of the environment under different circumstances. And that is why you are seeing a lot of fires, you are seeing a lot of floods, you are seeing a lot of winds and tornadoes. The other one I think is called Hidaya. All those things are happening because the climate is changing. That's one barrier. The second barrier is what I've just, when the Vice Chancellor was talking, I was fiddling with my phone because I wanted to get the current world population. So as at 11, and I think right now it's just 10 minutes after 11, the world population, that is a time clicking, it is 8.1 billion. That is from midnight up to now. So 166,000 people have been born, and actually 57 of them, 76,000 of them have died. What that means is population growth. And this has not included those ones who have just been born, probably in our villages. These are numbers that are ticking because they have in facilities that are already taken care of. What that means is that the number is conservative. Now, mix that with the emerging trend where we are realizing that the youth, Africa holistically depends on 65% of them depend on farming. And then the farmers themselves are aging. And the youth, and I see many of them here, are looking for white-collar jobs. So there is a challenge therein. Because the youth, they would want to TikTok fertilizers. And they want to weed using, Facebook. It can't happen. Your hands must go deep in there. It can't. So how do we then, and the thing is, and for those people who have traveled will tell you, my good professor, that some richest people in this world are actually farmers in the West. But then our responsibility is, how do we make farming trendy for the youth to actually go, if they are organizing alphabets for a girlfriend, for instance, to tell the lady that, I'm a farmer, and feel confident enough to declare themselves as a farmer. Because right now they would say, I'm an IT person, I am an engineer, but they are struggling to say, by the way, I'm a farmer, I eat IT farm, AI farmer, They are struggling with that. And therefore, we must think about making science and farming more interesting. Now, having said that, what are we likely to see because of those barriers? And there are many. We are likely to see something we are calling food and health insecurity. This is interesting. When you see such faces, and many of them are around us, then something comes in. There are opportunities that we need to take advantage of. And please pay attention to this slide. This is where it starts. This is the rest of the world. So this is a question being asked. What is your opinion on the shortage of food in the rest of the world? There is Arabia. What is Arabia saying? That's the question. Arabia's concern is, what is opinion? You see in the question, all they are spotted is the opinion. Americans are also asking a question. What is the rest of the world? Because it's America, who are these guys? Are there other part of the world? EU, in that question, is asking, what is shortage? I spent time over 4 or 5 years in the UK, and I can tell you, the food is in surplus. They have excess. So they don't understand what is shortage. And of course, Africa, we are also asking a question, what is food? So one question like that, and you see those divergent opinion, then something is happening. So please, if you have not got it, the challenge here is that you must know as Africa that you are on your own. Your problem is not shared. So work on it. So let me actually throw you to another slide, and I would want to see the reaction of on your faces. This is a slide. I want interpretations here. What does this slide mean? And please remove your imagination from horizontal acrobatics. Let's look at this. What does that mean? It's not about pockets. These guys, it's just a difference in interest. Everybody has their interest there. Remember, there's a couple, they are watching the moon. You know, this is romance. They're saying, wow, look at the world. But everybody has rested their hands in areas of interest. Now, while it looks comical, and probably you might imagine that it's probably a little, animated. I want you to think beyond that and realize that that's exactly what is happening in the rest of the world. Food insecurity is now an area of interest, and the whole world is putting their eyes and fingers at the right places for their own interest. Now, this is what it means. For Africa, we must stop sleeping. We are sleeping so much. And the sleep is that somebody needs to wake us up. Waking up, why must we wake up now? We must wake up because I want to take you through opportunities that we have lost as Africa because we are sleeping conveniently. The first one is here. In early days when there was generation of energy, the rest of the world used coal, which is COAL, to generate energy. And it's a basic science. For chemists in the house, what is the science around coal? The coal is used to heat the water. The water produces steam. The steam, therefore, is used to rotate the turbine. And when you put that within the electric field, electricity pops up. And the rest of the world for the years have used coal to generate energy. At that particular point, as African, we have a lot of coal in this country, and a lot of it is even in Kitui. But do you know we can't use it now? Why? Because it has been declared dirty energy. So who is compensating Africa for the confusion that has happened in the climate and our coal that we have not exploited? And right now we can't. What it means is we never picked the technology at the right time. So there's something about timeliness when technologies pop up. Second thing, the other one that we also lost, and I'm glad that colleagues are here, is called the Green Revolution. Green Revolution, there's a great man called Norman Borlaug. And there was nothing more special about that. The thinking was, and it started around the 1940s, it was just about high yielding, mechanization, and so that there is food production. Asia benefited, Mexico benefited, America benefited, India and Asia. Now is when in our villages we have tractors, and in other villages we are still using those cows. But from 1940 to now, we are talking about clearly almost 70, 80, whatever years, 80 years, actually 84 to be more precise, is when we are having tractors. What is interesting is that there is also another caveat. It is called carbon credit. So for the 80 years that they have been doing it, right now, before you export anything abroad, they are asking, what is the carbon footprint of this? So it's again a caveat. And conveniently to lock those who are slow to pick technologies. It is an orchestrated narrative that we must unpack as Africans. And therefore, we can't benefit from it. It's gone, I can tell you without. And you can see the drive right now is towards renewable. And I can tell you without a doubt, we are again being taken from our area of strength to an area of weakness. We have no clue how to make the lithium battery. So when will we actually benefit significantly from renewable energy? Who has the powers to do that? So when we are all boxed again to renewable energy for a dirty environment that we were never contributed to and somebody needs to pay us, we are being moved to renewable energy conveniently. So every time we are just close to get it, something pops up. And we are not learning. We need to change our thinking. Now, let me tell you, an opportunity we are just about to lose. It is called AI. artificial intelligence. And I'll bring it home. Here, a lot of discussion, there is this thing called ChatGTP. And because I serve in the management board of Kenyatta University as a registrar, and the CEO of this institution is seated here with the members of management, high level, the discussions around ChatGTP is a heated one. And you know how, again, we have been cornered? We have been cornered to think about it through the lenses of plagiarism. That, this student with AI, you just throw in the assignment into there and the results pop out. And it is very, very bad. At the moment right now, I don't know what is the position here. My personal position is that we are actually wasting a lot of time. We needed to have jumped into this AI so quickly and start looking at how do we build our capacity on it. One of the major problems, and we still teach, have been teaching until just recently when now I'm no longer teaching. But I can tell you the frustration of most of the academics is marking. It is a frustration. We've gone through that. Marking, teaching is the easiest and most enjoyable thing. You teach and the students get it. When you have 1,000 papers and you are supposed to mark them, and marking needs to be automated because as you are marking, it's subjective. It depends on whether you have taken breakfast, are you tiring, and there are error margins that if you were to actually factor them in, you will realize it's not very clear. So you probably need to automate marking. So what are we supposed to do? Basically, we set exams. Set your exams, and when you are marking, your reference point is that thing we are calling marking scheme, which is also content. Why can't you create a softer content of this your marking scheme? Create it as a reference point. Let the students also do theirs as a softer scheme. Create it as a softer content. You load it against your marking scheme. It runs on it and it will give you percentage similarity. And with AI variations, it's able to give you the variation, and they put a factor on it, and you can mark by a click of a button. All you need to do, submit, click a button. Then you even can allow the students to just use their AI and just type. They can use their laptops. Then the trick would be, as they are comparing this with your marking scheme, how different have you used your brain? Because that is the fear. They keep saying the brain is no longer going to be used. So we want your brain to look at those variants beyond what AI is giving. If you can make an anti-AI, which is, they made the ChatGTP, they have already made another program which is verifying whether that work was done through the ChatGTP. Already it's existing. Then they have already, we are talking a third one that paraphrases what has come from ChatGTP so that you are not able to follow. And we are here right now, they're existing. And I chair the committee for speech writing. And I can tell you there's nobody who can beat ChatGTP writing speeches. I can tell you, all you need to do is to say, what is this conference about this? Who wants to pick this? What is our strategic objective? That's what you feed in. After that, you give it 5 seconds. It will give you a decent speech. Your work is now to make it, domesticate it. You have saved that person's time in terms of thinking about alphabet to now domesticate it. We are talking, but what are we saying right now? We have been made to believe the ChatGTP is a bad thing. And we are actually locking it. In my own university, we have anti-ChatGTP. We will have that discussion. We are losing on it. The other one that now brings me to my level of confidence, this is what I do every day, is what we are calling recombinant DNA technology. Recombinant DNA technology is the technology that leads to what we currently see, what is called the GMO. And please, again, listen to me here. Let's now talk. And it's not difficult. There's a man here who can actually sequence for you these things very quickly, seated next to me here. Recombinant DNA technology started in the way 1970s. It's about the DNA. And this is the trick. What you need to know is that anything that is alive, be it a mango tree, be it a mosquito, be it a bacteria, human beings, all these things have DNA. Is the motherboard. It is the motherboard, and I will unpack that shortly. But every time when we say GMO, what happens? What do you think? It's usually very distorted narrative. And this is what it is. This is the GMO debate. If you are able to look at that picture, you will know what exactly I mean. The shell is the one that being cooked, the egg is on the pan, the fire is burning. So things are confused, and that is what happens in the GMO debate. Nobody understands it very well, and we don't want to also get to know exactly what this thing is. Why are we doing that? We have been boxed to think that we are happy and we are comfortable, but we are not. We are not. But when you look at that boy or that child, very comfortable. But this is misery. But this is what Africa is. We have been made to believe that sleeping hungry is okay. Tutakula kesho. We will eat tomorrow. It's not okay. And it's because somebody is wiring a narrative that there is no reason why, as it is right now as Africa, we should be having food insecurity with all this land and the best climate. So there's a challenge, and I will take you through the narrative that where we are right now, the reason why it is important that you understand the politics around this thing. We must appreciate that actually Africa is the big brother. Look at the younger population that we have. Look at the biodiversity that we have. The West are forever here with MOAs and MOUs to get the data, because what they only require is that you come here, you get single, DNA. When you go there, right now with synthetic biology, you don't, once you've gotten the direction, the rest you can synthesize. And it is okay. They don't need where the original source. They don't need it. So what is important here is what actually somebody called Isaac Newton reminded us years ago. And if for those who are doing physics, the third law of motion is action and reaction are equal and opposite. What it means is that any time I am talking because somebody somewhere has bothered to keep quiet. We are alive because others have died. And when there is drought, and I'll give you examples, when we are hungry here with those kind of empty plates, there is a child that is being overfed somewhere else in this world. One child with a whole chicken. When our cows were dying here because of drought, and it happened for 5 or so years, there were cows that were overfed in this same world. So the world provides everything. When it was drought here, it was actually flooding in Nigeria. Right now that it is flooding here, Malawi and South or whatever are getting higher suns, heat. So what that tells you is that we are struggling with our maize that cannot sustain the drought. And I can tell you, part of my PhD and the patent that I have is for stress-inducible promoter. There is a grass that is called resurrection plant that grows on top of the rocks. And it is a grass. And maize is also in the bigger family of grass. And we can't eat that particular grass, but we can eat maize. Then you ask yourself, can't we just move from maize to grass? Then you can't accept as human beings. The problem is our eating patterns as human beings is the reason why we are struggling. Because every time there is something that is edible, but we just don't like it because we have no clue whether it is safe, it has never been eaten, and it's the culture of food, and there's nothing wrong with that. There is no way, if you can't eat maize, we can eat sisal. Sisal will never have a problem with the drought because it's succulent. So, and it is available. Why can't we move eating? So that is where science comes in, and that's where we come in and ask the questions. Why is sisal becoming drought tolerant, susceptible, and drought tolerant, and maize is susceptible? What is in this? And all of them have the same alphabet, which I will explain later. What that then means is that we have a problem with things. Striga. We have a problem with aflatoxin. We have a problem with maize lethal necrotic disease. We have a problem with these pests. They are also, the pests, please, are also looking for just food. So when they are coming to eat your maize, they are also just struggling to eat food. But when they eat that, they are affecting you. So how do we coexist? When the world gives you, say, cassava, that's cassava. The worst thing with cassava is something we are calling cyanide. Cyanide is a defense mechanism for cassava so that it's not eaten. Please, there is a pathway called the Namarin pathway. My undergraduate was in biochemistry. What cassava does by producing that bitter thing is that it's defending itself from being eaten. So when you eat it, it's bitter and you run away from it. And the cassava that have a higher level of cyanide have bigger tubers because nobody bothers to eat them. So it is survival for the fittest. And I want to tell you here, if maize will know that that cob is what we are waiting for to eat, they will hide it on the roots. They just don't know that we eat them. And don't let them know. But with the time, they will realize, how come every time before we harvest our hands, this cob keeps going away? They don't even know it's human beings picking it. With the time, it will become shorter, shorter, giving you very small, and before you know it, it has picked out something. That is evolution. That's how it happens. Now, this is where we come in for food security. Remember, we are communicating technical research through this language. It's science communication. Anything that is alive, as biotechnologists, all the things that you see in there within the organelle at the cellular level, that is biochemistry. When you leave the organelles and go into the inside the organelles, like now you are in the nucleus, now you are going in the entire life of molecular biology is within that nucleus. It only plays around that nucleus and sometimes the mitochondria because it has a bit of DNA as well. But nothing else. That's molecular biology. So us guys in molecular biology, what we do is just to get the content of what is in the nucleus. This is what it is, so that it's very, very simple, colleagues. Anything that you see alive must have a DNA. And DNA is made up of only 4 letters. And I'm saying those 4 letters so that please understand your DNA. Your DNA right now, each and every cell has only 4 letters. And the onlyness will be qualified shortly. Occasionally, we have a U, but because I have a T there, I'm still fine with the 4 letters. So, the question would be, for carbohydrates, for instance, the building block for carbohydrates are monosaccharides. They join themselves. For proteins, the building blocks are amino acid, and there are only 20. They join themselves, and a few derivatives. They join themselves. For lipids, the building blocks are fatty acids and glycerol. That's all. Nucleic acids, which is what a bigger name for genome and the DNA. People talk about DNA as if it's the only. There's also RNA. But together they are called nucleic acids. Those nucleic acids are only having 4 letters. Those 4 letters are A, T, G, and C. Adenine, thymine, guanosine, and cytosine. Just know them that way. Now then you will ask me, if they are only 4, how come that this is an elephant, the other one is a horse, penguin, and all this? How come those 4, they should give us the same living organisms? And that's a good question. The variation is in terms of the sequence. How are those 4 letters, A, T, G, C, following each other, and in how many, what sequence, and what numbers? For instance, in your DNA, you as a human being, you have 3.2 billion of ATGC. So it will, ATGC is a start codon, ATGC, and then they say ADT, whatever, until 3.2 billion of that in double-stranded. And it is folded in the nucleus. So others will have less, others will have more, others, and that is where the variation now is. And this is how it follows itself. This is just to show you. That's how they are connected. So between one T and another one, there's something joining it. And I don't want to go into this, the technicals, because that's not the essence of today. The essence of today is to understand this. Now, I want to break it down for you to understand. For instance, look at your DNA as alphabets. Alphabets, we only have 26 alphabets. And if I want to, all of us have picked our names from those 26 alphabets. So if I pick my Vice Chancellor here, whose first name is Thomas, for instance, and I tell him to write his name, you know he has 26 alphabets. But I can tell you, when it comes to writing his name, he will be running around, he picks where T is. He has only picked a T, and he has left an S. He goes back again, looks where H is, he picks H. Then he goes and picks where O is. At the end of it all, his last picking is S. He has pulled his name out of that as Thomas. Now go look for another gentleman sitting closer here called Philip, our DVC. He will again go pick his P and H and all that. He has pulled again. Remember, there are just 26. So if you go and pick my name as Richard, now I go pick. So we have 3 names. So please understand this. We have 3 names. All of them have been picked from 26 alphabets. And if I tell you now, write Joyce, there is no way you will pick Philip and pick Thomas and pick Richard, and from those that have already been created to make a Joyce. No, you go back to the alphabets. Now that is what the DNA is. DNA, once, and that is the transcription, once it has created something, that becomes a species. So the Richard becomes a species. So Richard for Richard, Philip for Philip, and you cannot mix them at the species if the species has already been separated. That is why every time we go back to the genome when we want to pull the DNA. Now listen to this. The letter alphabet can write a letter. And depending on the content, remember it's the same 26 alphabets. Depending on the content of that letter, if it's about promotion and bonus, then you will get this reaction, isn't it? From the same letter. But the same letter, from the same alphabet, you can actually pick it and give a sucking letter. You will see, you will get that reaction. So what does that mean? That these alphabets, 26 of them, is very interesting. Do you know certain things don't make sense until when they are linked to you? For instance, if you come to your office and allow me just to use this, not because of any other reason, and you are told, and because my DVC, and you get a note written to you, it is now your turn. If the next thing that follows, it is your turn to become a CS, no. It will trigger something else. But if it is your turn and the next thing that follows, you are told, you know, there have been plane crashes around, you know, these things happen, you know, what will it trigger? It is the same thing you are told, you know, and it's your turn. So what that means, it's the same language. It is your turn, but it has different impact. And that is the translational bit when, after you have gone through the central dogma, I'm trying to break down the central dogma, that the source of the DNA is copied to an RNA, messenger RNA, and then translated to a protein, and the protein is converted into a trait. And this is how it happens. And follow this. When I give you a CD, when you can't put, even if you put it on your ears, you will not listen to the music. Why? And yet it has the music in there. You will require that radio cassette to interpret the content of that particular CD. And then if the music comes out, that's the protein. If you are expecting a gospel music and you listen to, you get that it's now reggae. Where do you go to? Do you beat this radio cassette? You go back to the CD, you start looking, which one did I pick, by the way? Meaning you're going back. And at the end of it all, depending on what music, you will be dancing. That is a central dogma. So this is the trait. So what then this means is, let me tell you how we have used genetic engineering and in medical field. When people tell you GMO is very bad, please listen to them and ask them a few questions. Number one, do you know that there is a drug called Zimap? Zimap is a drug for the Ebola. It was actually expressed in GMO tobacco, transgenic tobacco. And it was actually allowed by WHO to be used because of the crisis of Ebola before it completed its clinical phases. How was that allowed? And nobody was there anteing. Then there is something called insulin. Any person who is diabetic, and with a lot of humility and respect, you are enjoying GMO insulin. There is no non-GMO insulin world over. And you've never heard about that. Why? Because that technology is being sold from somewhere else. Before this technology of recombinant DNA technology, we were killing pigs. You know, pigs and human beings are closer. We were killing pigs, you purify their insulin, and that is what we were injecting on people. But now if you have, fair enough to yourself, you realize that what were we doing to our Muslim brothers? Because they can't touch that your insulin from your pig. So they were dying without it, and the insulin was there, number one. Number two, also some people are developing some allergies and reactions because of some things, the epitopes that were not matching. Then the recombinant DNA technology asked a very simple question, that human beings, there are people who are not diabetic. So what have we done? You go into their genome, pick that section of the DNA that is responsible for human insulin, and then move it into a cassette, then express it in E. coli. Then target it so that it is an extracellular waste. So the bacteria doesn't know that it's producing actually insulin for you. It actually thinks that it is just excluding some waste out there. You purify, and now you are using human insulin. That is GMO. How come we are not hearing that story? Because the discussion has been skewed. There is also something we are calling Gusher's disease. Gusher's disease is a disease of regeneration of the bones. It is being one of the drugs there is being expressed in transgenic carrot cells. Those are transgenic. Nobody is talking about it. Then recently we talked about the COVID. Are you aware, and I worked for Pfizer, and Pfizer was the one that giving you those vaccines during COVID when everybody else was running up and down. Are you aware that it was the DNA that was being injected in your system? And let me ask you, if you think that that is very bad, when you are eating, and many of you are going for lunch, and I will be monitoring if that lunchtime finds me here, I want to see, when you are eating, do you usually bother to go inside the nucleus and remove? Do you know you are eating the maize with its DNA? When you are eating beef, are you aware that you are eating it together with the DNA? When you are eating beans, you are eating them with those letters. Why haven't you ever woken up with the leaves because you ate beans? What happened to those DNA? And in your mouth, when you are eating ugali together with the beef, you are eating DNA of beans inside the mouth, DNA of, if it is chapati, of barley, of wheat. If you like frying with tomatoes, DNA of tomatoes is in there. If you want some other, so by the end, when you are chewing those things in the mouth, your mouth is genetically mixed organism in the mouth. It is genetically mixed. And you don't wake up with all these things. So why are you eating beans and rice? It is because you want the proteins from beans and carbohydrates from rice. Very basic. And this is what genetic engineering is doing. It is telling you that now there is flood that beans cannot grow, but you still want proteins. So what do we do in genetic engineering? All we are saying, naturally you eat your rice. Naturally you eat your maize. But now maize doesn't have capacity to produce proteins because it is being produced by beans. Beans and maize are both plants. In your mouth, you eat them, you don't get sick. So what do we do as genetic engineers? We come and sequence the beans and identify that those letters, A, T, G, C, those ones, that is responsible for producing a set of proteins. That is what we cut. We move it into the genome of maize and allow the maize now so that seed that you're going to have has the original carbohydrates, but now has the same proteins that you have been eating in your beans. Imagine at that particular point, that maize is called GMO, and it is so bad it causes cancer. It is so fast, and this is very important, that right now, when you are eating beef, you are eating the meat part of it, and nobody eats a cow that is walking. Cows, by the time you are eating it as beef, it has already died, therefore it's the tissue. This is the question that is happening in what we are calling regenerative medicine. Why must you hurt a cow that is walking? Why can't you leave cows to be for optical nutrition? You just look at them and you enjoy. Then the cells that are growing inside the cow, cells are known, and we know the pluripotent cells, and you create an environment that is like the cows, and then its work is just to create for you blocks of beef. It is already happening in Israel. So leave cows alone, don't hurt them. Because when you are eating cows, you are eating dead. There's no cow that is eaten alive. So this is where science is going. But when I tell you so, you start wondering, hey, is that still a cow? By the way, are you sure? That's a cow. It is still the cow. You have sequenced it. It's still the beef. And that is why the West have taken advantage of our rigidity and caution to take advantage so that you don't pick the technologies at the right time. Why? So that eventually they make it and they will sell it for you. Those tissue and regenerated tissues will come here as a can, and we will be buying them. And when before that, we are still saying, this is not looking good. Listen, what the anti-GMOs are telling us, they are saying that we go organic. Truly, if we must go organic, this is how we should go. Leave alone or whatever thing organic means, we should go back to where we started, hunters and gatherers. This is the right organic. But the moment we started now doing domestication, we started bringing biotechnology. And let me tell you how ready we are as a country to enjoy this GMO foods. And I will explain. One, all the countries that have already developed and picked GMO must have these things that I'm about to say. One, they must have research facilities. They must have human capacity. And I can tell you, they must also have what we are calling regulatory agencies. They must have an appeal board and the government goodwill. All along, in this country, and we have a ban on importation and whatever for the last 10 years. All along, we have been struggling with the government goodwill. The research facilities are available, and I'll take you through just a few of them. Capacities, the kind of PhDs I've trained, and the lab, the institution at Kenyatta University, we have the lab from 2005. You know, when you talk about GMO, you think it's coming from the West. From 2005 to now, those are 19 years, we have been making GMO at Kenyatta University. And regulatory agency, we have the National Biosafety Authority that actually you need to know that is one of the most competent institutions we have here. The first CEO was Tonui. That was a PhD holder. The second one was Professor Ogoi, PhD from University of Nairobi. The third one right now, Roy Mugira, PhD, very eloquent people. It's almost that you must have a PhD to lead that organization. Their work is to look at and regulate anything. And let me tell you, the moment a government decides that we are setting up a regulatory agency, it means that the government has already accepted the technology, only that it wants it regulated. There is a reason why we don't have cocaine regulatory authority. Because cocaine is banned. And therefore it becomes a responsibility of the police. We find it with you, we arrest you. You ask yourself this question. Why is GMO banned in this country? And me, as a molecular biologist, we are in the university being paid salary to train students on genetic engineering. Is there somebody training people on how to smoke cocaine and being paid money? There's none. So you ask yourself, where is the confusion? And people need to understand, where is the politics around the GMO? It is never about the science. It is about who benefits, and it's not about the safety. Please get it correctly. There is no way American government, the first GMO was commercialized in 1994. For the last 30 years, they have been eating GMO because they know they are eating just the biomolecules. But here, 30 years later, we are still having a ban when the technology now we have moved to genome editing. So we have not again benefited from that first generation. We are not benefiting. So what it means, and please this, I'll move a little faster now. Kenyatta University is the only university that has a plant transformation laboratory and was the first in the country. Thereafter, there was Calro. And then there is ILRI Becker, the Becker then, and ISIPE now. So any other person who does genetic engineering either comes to KU, ILRI, Calro, or all those places. And that does not mean that this university doesn't have molecular biologists. This, and I even know Jen Binot, or those were my classmates and whatever, they know how to do genetic engineering, only that they do it in those labs, because those, for you to have such a lab, the NBA, National Biosafety Authority, must come and verify. And also, remember, the genetic engineering is a small point of the whole process. You can actually make your construct here in this university. It's still not GMO. The only GM bit is when you're now moving it from that construct now to put it the transformation. That small bit only is what requires that regulation. And thereafter now how it happens going forward. And just a quick one. Now look at what is happening at ILRI Becker. Cassava, banana, plantain. Lena Tripath is leading that very heavily. In ISIPE, Drosophila, just to understand how animal GMO is happening. In Calro, a lot is happening. And for your information, Calro has a lot of also trained personnel. A lot of it. And there are a lot of things that they have been doing with CMIT, AATF, Bayer, and Pioneer. And there is cotton, BT cotton, the one that we are talking about, is already, it's a GMO cotton. It's already in the field, by the way, for your information. The GMO is in the field, but there is another ban. So you get the confusion. Huh? The GMO cotton is already in the field, but there is a ban on importation and consumption. And then you wonder, when you see cotton, this is the fallacy. When you see cotton, people think that cotton is for the fabric. No. 60% of the cotton, actually, people who do cotton, they don't do it for the linen and lint and all that. It's actually 60% of it is for oil, for the cooking oil, and the other, whatever is left, is for animal food cake feeds. So this one for the cloth cotton is the smallest, and actually they don't actually grow cotton for that. But in the country, and we like, when we sold it, that it is for manufacturing, it saw the end of the day. That's what we did. We said it is for manufacturing industry, Agenda 4, and it worked very well. And then it was released. But the 60% of it is actually food. Cassava is happening with Miano. Actually, it's about to be done. At Kenyatta University, this lab that I have headed for years before now moving up the ranks, is actually doing genetic engineering, and we have done all those crops you are seeing there. We have been genetically modifying them at Kenyatta University. Have a lot of transgenic seeds in the lab that needs to go to the next phase. And these are the kind of things we look at: disease, insects, weed, drought, salinity, and others, and now aflatoxin and many others. And this is how we write the genes, but don't bother about this. This is not important, just to let you know that these things happen. So I'm taking you to understand that we have locals who are doing this. We are so good at this thing that we are training for the region. The first one was my PhD student. That was from Tanzania. Tanzania gave them money to come to KU so that we train them through the Asareka funding. He graduated and went with his seeds. The second one is Leta Tulu from Ethiopia. Graduated and left. The third one is Russia from Sudan. Graduated and left. And I'm picking that because of the competencies for even training for the region. There are so many that are happening. This other one is Jonathan. He's in the US postdocing. He was in IITA recently. What he was doing was actually, he's making drought-tolerant maize. The other one is Eric. He's for Meru Chuka University. And remember, the upper one, so that you understand the networks, that one with University of Cape Town for where I was. This one is with Eduardo, UC Davis. The lower one with Poland, that is with Dorota. The other one now with Belgium, Ghent, is the source of all this transformation and the plasmids. This other one with Nevada. And when you go further, this is Aptama. Aptama means that, imagine, and please understand me, if you give birth to a child and you still take that child for injection for vaccination, is it because you are doubting that God never completed his work? Why are we vaccinating? That God was not perfect enough to create us a child that we don't need to vaccinate. And we are saying if we are comfortable with vaccines to vaccinate our own kid, malaria, all these things we do vaccination for, you think we can't vaccinate maize? Maize. Maize. And human beings we are vaccinating. And now maize, so that stem borer does not eat it, so that it grows and gives us, some yield. You think that's what Aptamas does. And this is maize-struck virus, and I can go deeper into that. This other student, Joel, was mine for aflatoxin. He's now in Penn State, after graduating last year, June. We are so good that the Penn State took him as a postdoc right from my lab. Chop. It's because the knowledge is so basic, we will tell you, we know those letters, and the gene here that we can tell you this is a gene here is the same in the US as well. And this other one, now he's gone. And you may not get him. Professor, that was my second student, that's Wilton Mbinda. He is actually professor at TUM, Pwani University. He was actually looking at the drought tolerance. This other one with the Germany, Nzaro, is in TUM. Actually, the whole thing that he was doing is that we are realizing these trees never go drought, they never dry even if there's drought. What is the reason? It means the roots are so deep that they get some water there. Then we ask ourselves questions. Why can't we elongate the roots of the maize? Must it have this height? Can't you make it 1, 2, 3 meters? What is wrong? Because we don't eat the roots anyway. And roots exist here. Why can't you pick? Because there are plants that already have longer roots. Take that section of gene, put it in maize, target it to the roots. So that the roots, if it is 3 meters of roots, you will never find drought in maize. Because at 3 meters down there, there's some softness. And these are the things we are talking about. And we must simplify this research to a point that we appreciate it. So we are talking about striga again. Striga is the most notorious. It's a parasitic weed. It has everything, every machinery to produce its own food, but it wants to wait for maize and sorghum to produce food, and it sucks away. And do you know how it germinates? There's a lot of chemicals, the stagolactons, and it's a very interesting thing. So what have we done? We are saying that striga does not attach itself on a mango. Why? Because mango has ability of something we are calling purple acid phosphatase. It's able to deal with that. And that's Prof. Runo. He's the one running with that. We have Neondo, that's a staff member of JQuat. So right now, if I have to list, every institution has actually passed, has a staff that has passed through that, the plant transformation laboratory. Even in this one, there's Jane Binot, who studied, was my colleague for master's, he's here. Just colleagues, and doing a good job here as well. And we have things around maize-streak viruses and many, many other things. And a lot of GMO is in the field. So when you hear GMO, don't think it's coming from the West. It's already here. These are the areas that we are testing them locally here. And now somebody will start asking you, these are the questions you usually get. Why is Africa running very quickly? What is the speed? Why are we going there? The thinking is so that you know, In Africa, as we are talking right now, in the southern part of Africa, South Africa picked GMO in 1998. And I spent 4 years in Cape Town. So I was eating GMO, and I'm still waiting for those leaves. There's nothing. 30 years right now, people have been eating GMOs. Those leaves are not popping up. So in South Africa, we have South Africa, Eswatini, and Malawi. They have already commercialized. In East Africa, we have Sudan, Ethiopia, and now Kenya, because we have all. In West Africa, we have Nigeria and Ghana. Now Nigeria contributes to 25% of the African population. 1 in 4 Africans should be a Nigerian. And they have already picked it. And when they picked it, when we are still debating here, they are now the world leading exporter of GMO cowpea globally. But here, we are still debating. So we are training for the region. Then the question would be, sometimes after all these things I have said, good people, I ask myself, why are we here? At what point, where did we miss the point? And please tell me what happened here. Is it that KPLC came in and put the power, or this person built, because this is a real house. Where did we miss the point with this research? And if you ask me, the point is not very bad. The challenge is we have allowed as researchers of this thing, because right now you are hearing this GMO, you think it is new. For the last 20 years it has been happening at Kenyatta University. It means that we haven't done a good job as the researchers as well to speak about this matter. And for that reason, what we are seeing out there, you will be told that this is GMO. So anything that seems big is GMO. Please listen to me. There is no GMO chicken in this country. Those chicken that you see, they are rotating here, and you call them kukupono, you know, for you people, that they are rotating here without clothes. Those chicken that you see, what has happened there is, if you are to sell me on the basis of my weight, and I'm doing business, how do you feed me? You bias the feeding. In the morning, you give me 37 sausages. I eat them, and you force me to eat them. At lunchtime, like right now, you give me a liter of cooking oil. You see, I swallow that one. In the evening, pork, 2 kg. Within a week or two, I will be having very nice size. Then you weigh me and sell me. But you have not touched, at that particular point, I am not a GMO. It is just, that's not GMO. It's just the feeding. GMO we talk about when you touch the DNA. That's not GMO. That is a feeding profile. So the growth hormones that they are putting, they are lacing their animal feed and chicken feed with, it is for purposes of giving them quicker growth. So when you see that chicken, eat it. But what it has done, it has given you a bias, a certain level of amino acids, which when the cells get them, they know what to do with them. They will organize them into proteins and place them in the right places. And it is your business, because the cell is taught not to be wasteful. That is why if it finds more glucose running in your blood, it makes it into glycogen and throws it into your liver and others in the muscle. You will deal with it later. If it finds that you have given it a lot of fats, it will convert it into triacyl glyceride, throw it into your adipocytes. That is why any time under adipocytes for men, it's a lot more under the belly. That is why you ask yourself, how come any man who wants to become a little philanthropic starts from the stomach and not here? Why can't those fats go to our biceps so that we look good? Why must it start here? It's because the cells called adipocytes are largely under the tummy for men. And that is where the fats are taken. So you struggle with it, but we keep giving it to you. That is not GMO, good people. That is just a biased food. You see this thing, you say that's GMO. And then the worst bit, they tell you, this GMO people, you know, they are injecting things in the labs that we don't know. Let me tell you the truth. If it is true, and this man knows those, and many people here know it, if there is a genetic transformation in this method, then do you know what that means? That you eat maize, you grow maize. You become maize. If this is that easy, and the DNA itself is semi-conservative, meaning it is stupidly protected, that any new gene or any new sequence that once pop up is knocked off. Proofreading mechanism. So there's no method like this. But the anti-GMOs know how to trigger the emotion. These are their photos. These are not mine. These are their photos. They keep telling you that, you know, these GMO people, outside it will be apple, inside it is orange. I don't trust them. You know, and they speak very quickly and you will believe them. Actually, they will tell you, look at the way they have played with that. They tell you that is a pepper here on pilipili hoho. What they have done, and this is serious art, they are telling you GMOs can be happy outside, but inside they are crying. You know, they are crying. Look, they have a way of actually communicating, and the worst bit, they tell you that the GMO causes cancer. And let me tell you, and I don't know why they picked cancer because they think it is the most scary disease. But if it is true that the GMO causes mutation, why can't that mutation cause blindness? Because there are a set of genes that are creating vision. But they know that we have spectacles and we have ways of dealing with those errors, so that's not worrying enough. So they have picked cancer unnecessarily. And people who actually keep thinking about that, my own mother died of cancer. I get very emotional around cancer, and I cannot be leading research that leads into this. And we live in this country. These are the things you have always heard about GMO. And please, if yours is not here, add it. They tell you and ask me those questions because I should be able to respond. GMO causes cancer. You've been scared to death. GMO seeds do not germinate. There is no such thing called GMO seeds do not germinate. The thing they keep talking about is acetanose terminator, which is a stop codon in the transmission, in the formation of the cassette. But it doesn't mean, and this is why sometimes I wonder, if somebody says GMO seeds don't germinate, why can't you just tell Oduor, you have GMO, do you have them? Yes, bring them. Media, let's plant these seeds here in a lab. It just takes 3 months. We will have seen them grow, yield in the timed media. Then we plant again, and then we stop this discussion. Then you see that they really germinate. The only reason why the people are told not to, to plant the same seed is the same reason for the hybrids, that it loses the hybrid vigor, but not because of germination. No, they germinate. And the attempt of that technology of stopping germination was actually also triggered by the antis. Because when the regional stages, when people are saying that the pollen grains have moved and all that, so they were trying and make that die in the next phase. They are the same people who came back saying, no, you want to control the seed. But I can tell you, because I have a lot of some stuff going on around cancer, this terminator gene is what we are now researching for cancer. Because cancer is about uncontrolled cell division. So if one cell gets that particular problem, and then you have the antisense for this and stop it from cell dividing, then we are actually using it the right way. Then they tell you GMO cause impotence. You touch about impotence for men, you have already taken us away. We won't touch it. And they have picked it. They tell you, do you know that it causes infertility? And this is where I get a little worried, because infertility, the kind of research that is going on in pharma industries where we have worked, if it is true that GMO causes infertility, then I can tell you we will not be selling the GMO in the market. We will be selling it as a drug in the pharmacy. We can't, and it will be so expensive, because the kind of money that goes into the research for contraceptives for women and for erectile dysfunction and all these things for men, we will be using GMO. Once you have had enough of your kids, you start eating GMO because it is making you infertile. Yeah, we will use it as a drug, and actually we'll have another name for it. So these are stories that people tell you, and you listen. And then they tell you biodiversity. If you are reducing the number of spraying because of BT content, it means you have actually given opportunity for more pests that otherwise you would have sprayed to kill, in that particular. And then they tell you GMO is equal to mass national. I don't work for Monsanto or Bayer. No. And then they tell you who is funding GMO. Then I ask them a question. Even this very, very country is getting funding from China, from EU, and from the US. Have we become Chinese? So why does it become a discussed story when it's about donors like Bill Gates funding research, when the entire country is actually getting funding from China and all? So they are destructive stories, and it's sad. And all these grants I'm talking about here, like half of them are actually funded by NRF. NRF has funded us to the tune of over 100 million right now. Even right now as we are talking, we have grants from NRF running, and that's a government funding. So what I'm trying to say is this. Please, the discussion around GMO is usually very polarized. Everybody has their side of story. Like now you don't know whether this person is looking at you or the other side. And when you believe he's looking at you, that is your belief. When you think he's looking at the other side, that is his belief. The people who believe in this technology believe in it. Those who don't believe also don't. And you can do nothing about it. What we find very interesting is that every time we are making progress, we find a problem. Like this government came in and said GMOs can now whatever, and are you aware that we are in court as we are talking right now? So we were too close, just like this road. This is a very nice road, but look at where the direction of the street lights. You are very close to it. Then something very small is what is, now we are in court. There were 7 count cases. We have collapsed them into 4. Now we are still in court. Yet the government has already said, this is okay. We have the systems, we have everything checked, and we are happy with it. And half the time, we as the scientists are very, very, very misunderstood. Very misunderstood. And I don't know where this misunderstanding comes from. This guy is the scientist. He is busy in the lab doing his thing. But I can tell you he's going to get a slap shortly. Because the lady is expecting a proposal. The person is just tying his shoelace. This is a case of misunderstanding. And I keep wondering, how did we go wrong as scientists? And I want to tell you, the fact that the GMO are new, the fact that it's new, does not make it bad. Please, if it is new, this is an innovation. It doesn't mean that it is now bad because you have never interacted with it so far before and somebody will tell you. Really, this is a serious innovation, but it will not fly in my village. Because the hair is perceived for superstition. You can't touch my hair. This innovation that carves the head is not right, and it will be evaluated. It is new, it is an innovation, he's not spending on the cap and everything else, but because it is new, it is bad. And that is what we keep saying. Not all new things are bad. The latest something, there was a time when the people who were growing chicken decided to have featherless chicken. And let me tell you the story. Featherless, who amongst us here eats feathers? Chicken feathers. Or, if you eat, you can tell us so that we break it down for you to eat. Nobody eats it, actually. Nobody eats it. So the feathers are just the other version of the clothes for the chicken to keep them warm. But the current chicken that we are having are so lazy that they are in rooms that are heated. If you have reared chicken, the current chicken we are wearing, they don't need those feathers. So what have we done? The scientists have said the gene that is responsible for feather development is a gene. You silence it so that the chicken can just roam in this house, nicely in the heated environment, so that because when you are going to eat, to harvest the chicken, you must remove the feathers. And for some of us who grew up in the village, the process of having a chicken prepared is very emotional. It starts by your auntie telling you it is that chicken. Then, once you have been told that's the chicken, you prepare yourself for a decent, bonding session, with it. You chase it until you get it. And when you get it, you put it down there, you step on the head, you remove some feathers, you cut it, you tap the blood. The blood must be tapped because it's the sweetest part of the things. So you understand, that means, and some of you probably you are not aware, that's how we grew up. Then you boil water, you remove the feathers. So the bigger companies that are in the chicken business realize that they are employing so many people for manual removal of feathers. So they come up and say, I need the chicken without the feathers. But that, much as it is a good technology, it won't fly. In the Western, in my area, we won't, because removing the feathers is also bonding session for us. So you must also understand when the technology is made, is it speaking to the cultures of the people? And that is why it's not here. So I'm asking myself, we must accept that this technology, we can coexist. Here, regardless, if that guy pulls that trigger, he's also dead. Yeah, nobody's safe here. So they have to talk. These two must actually talk. And imagine they will. Because of the situation. This is what we are saying. There's nothing wrong. And by the way, get me right. I am not doing this research so that you eat GMO. If you have your organic and you like, it is also okay. From where we sit, we are saying, allow scientists to do the research and even keep that GMO genetically tolerant maize, which is tolerant to drought, so that when the drought comes and you have tried all these things and it's failed, you have your own. So that it is a strategy. And that is why I'm here to tell you, we need more of champions. And you must step back and ask yourself questions. Honestly, what is this GMO? Is it this really bad? So you must stop this fire that is burning. And for you to stop the fire, it will come with repercussions. People will call you, you are working for Monsanto, your money is coming from whatever. Like this monkey, I don't know what will happen shortly. But the fact is that it has gathered itself. Many of us, we are forever in the television talking about this GMO story. And people would think that you are working for Monsanto or Bayer now. No, we don't. We just like the science and we know what it can do. And what the anti-GMOs have done is that, you know, they have repeated lies so many times that it's looking real. Like anti-GMO will tell you that actually that is the shadow of this man. You know, they will tell you that that one is the shadow of this guy. And it looks, by the way, if you don't bother to ask the right question, you would actually think it is true. But it is not. You can say it so many times, 1,001, it is not true. That is not the shadow for the guy. And that is what we keep saying. Please listen, take time for science. And as we are debating here, the rest of the world are running with the money. And as they are running with the money, mine is to say that at the end of it all, we start looking very skeptical. The problem that we have as Africans, we are saying, hey, are you sure? We are seeing girls and boys and whatever, the bodies are changing. This must be GMO. GMO for the last 12 years, since 2012 up to now, there has never been a GMO in this country because there has been a ban. So all these cancers that have been happening, where have they been coming from? If it is the only source of that. So it is a story. And if you're not careful with this skepticism, it becomes very dangerous. You know, because you now start looking at, we don't want the West, you become xenophobic, you don't want the newer technology, and it is really so wrong. What is my belief? My belief is that we need to be very innovative. You know, this is a photo of a lady who was told by the husband, please, are you guys happy at home? Is everything okay? Then she send me a photo of yourself. Then she sent this photo to the husband. Are you getting that message? Yes. It means, dude, we are okay shopping. The fridge is empty. The fridge is empty. It's just allow. So how innovative are we for making innovations that are unique to us? And not everything don't have solutions. No. It's not like we don't have solutions. We can have solutions, good people. We only need to look for them, seriously. And all we need to do is to make sure that we are confident enough about our abilities. You must be strong enough and believe in yourself. That tuk-tuk is more than many of us scientists. They said, I will carry this. You know, so how bold are we in the areas where we are doing research? And it doesn't matter. Just do it. It doesn't matter the audience. Just do it. It could just be one person who believes in what you are doing. Just do it. The audience doesn't matter. Because if you continue doing that way, then you will be changing the narrative. Because for us, we believe that GMO, if read properly, the other way around, it is, oh my God. And that is what we believe in. So we need to change the narrative because that is important. Allow me to appreciate the gate that we have at Kenyatta University. That is our administration block. And without further ado, thank you so much. Good afternoon.
Let's appreciate our speaker once more. Thank you, Professor Oduor, for the informative and exciting talk. I believe all of us have gotten something. Because of trying to manage time, we will entertain a few questions, and this discussion will continue even during lunch hour. I know there are people who are following us online. We'll also get their questions, maybe in some written form, and then we can share with the speaker and see how they'll be able to get the feedback. So I'll take maybe a few questions, 2-3 questions. We can see Dr. Achono down there. Okay, Dr. Barasa. One more. So that we take the first 3 and maybe the rest will have some discussion later during lunch hour or tea break. Okay, so we can start, Dr. Achono. The floor is yours.
Thank you.
Be brief and clear.
Thank you, the moderator, and good afternoon, everybody. I want to appreciate so much the presentation from our keynote speaker. I want to appreciate the passion that he has on implementing modern technologies, innovations that are coming up. My concern with Matters West is embracing the technologies that are bringing up. When you go back to those days when you talked about when you are drawing blood from that chicken, those days people were not dying as they are dying today. The death rate, the longevity of life for those who lived those days were very excellent. The West themselves are running to, they call it organically produced substances. Simply because they recognize that going the direction of innovations, things are not working so well. The death rate, the obesity, among other things, is becoming an issue. What is your take on the West running away from their own innovations? That is one. Number two, what is your take on the technology of implants that are being promulgated to identify people wherever they are? That is coming from the West. That the children are going to be implanted with chips that can identify them wherever they are. These are technologies that are coming up. When you are embracing technologies from the West, what is your opinion on these two? Please. Okay. I hope I'm clear.
You're clear, Dr. Barasa?
I'd like to appreciate the keynote speaker for the informative session that he's given us. I mean, I think we've learned a lot, and he's really crushed it down into piecemeal pieces that are digestible. Personally, I'm a GMO freak, although the professor is my personal friend, but he knows that I'm a GMO freak. I don't like GMO. Although today I'm sold. And the only suggestion I have, and I think he might need to pay me for this very insightful opinion, is that yes, we are skeptical about GMOs as Africans. But I think the way forward for this is to Africanize the genetic GMO debate. If we, for example, if we take the genetic material from our own seeds, from our own seed banks, for example, we've got these indigenous crops and indigenous seeds. If we take the GMO, if we take the genes from that and then transfer them into our own, into other maybe products like maize, as you say, then that will go a long way in convincing us that the genes that you've taken from the genes are not foreign, and they are just African. So if we are able to Africanize our genes, then I think most of us will be happy to adopt that. And thanks for educating us once again. Thank you.
Okay, do we have the third one, and then we'll allow the presenter to respond to the questions.
Good afternoon. Yeah, I have this question to Professor. We have had many entertainment and facts about the benefits of GMO. So I'm requesting that he may touch on the disadvantages that GMO has.
Okay, Professor, maybe you could respond to those three.
Thank you. Thank you, Steve Baraza. That was my colleague when we were doing postdocs in Pfizer together, so he's a long-term friend. And I know he's a GMO-free, and I'm very happy that he has started warming up. Thank you. The other question is about the organic obesity and how come that the West are running away from their technologies and all that. In 2013, I had a privilege of spending time with Novartis in Boston for some time. And one of the drugs that was being done there, right from Pfizer, when we left there, Steve, was the drug that was supposed to deal with the obesity, because the problem they've realized with the Americans and places where foods are in abundance, it is nibbling. That each and every time they are eating something. And that's where the words cookies, the nibbling and eating. If you know us as Africans, you know the times for the eating. It was first of all, you wake up in the morning, you go to the farm. Farming was now exercise. So before that, you come home, before you go to school, you take some porridge, if you are lucky. Then you dash to school, you wait for lunchtime. You come, you eat lunchtime. Between lunchtime up to another time you are eating, there was not this 4 o'clock tea was never our story. We eat in the morning, probably at lunchtime, and in the evening. And the cells are supposed to do so. The challenge with the West is not the obesity. Even us now, and let me tell you, if I feed you on organic food, and you eat it in that speed, you will still be obese. So organicness or the GMO-ness is not the thing about the GMO. This is what they are doing. Organic is a swag for the West. Not everybody is eating organic. And what is organic? Because we need to break it down, and this is where we go wrong. What is organic? Does it mean that now we have new proteins? Does it mean that we have new monosaccharides that the GMO comes with? Let's go theoretically, scientific a little bit. Does it mean we have fatty acids that are fresh that GMO creates? There is nothing new that GMO creates. The biomolecules are constant. When there is somebody who is giving you a lot of biased food, so the West, the drug that, and let me take you that so that I respond to your question, they are starting to look for the drug that slows the appetite. So that when you take this drug, and at that particular point it was about when you take it, you don't feel hungry, and therefore you don't eat. And that way it gives your cells opportunity to metabolize and crush whatever you put in. And in that conference, I told them that this drug will seriously sell in Africa. Then they were like, why and how? Africa, there's not enough food. I told them, yes, we will still buy this drug because we don't have the food. When it gets to lunchtime, I give my child. They will eat that for 2 days without food. And the 4th day, because by the way, you can comfortably live for a day without food, and it is okay. This feeling of hunger that wants you to eat lunch right now, you can comfortably survive on whatever you ate yesterday, even now. But that bit around hunger is what is triggering you to go for lunch. The body has enough, trust me. So what it means is that the obesity you are seeing in the West, and the obesity is also here, and we don't eat GMO, is a cultural variation and appetite that is not guided, and the biasness of the processed food that they are eating. Processed foods are not GMO. Even the organic one you can process and make it something else. So what is really organic? Do you know GMO seed can be grown organically? Because what is organic farming? It is about get the seeds, use organic manure, don't add fertilizer, don't spray. That's the organicness. There's nothing so special about organic. So if I have a seed that is GMO that I'm not spraying on, I am not, adding certain chemicals on it, there's no fertilizer that is synthetic. Is that organic? Or the organicness starts at what particular point? Because you have mixed the DNA. So that's a discussion. I had it somewhere else, so we can have that discussion. Running away from the innovation that is their own is a problem. It is about when the technology is not working, you move to something else. But us, it is not about picking what is in the West. And I like whatever he's saying. The implant discussion, personally, having spent some time in the medical field and now in food security, ask anytime there is a new innovation, this is where we go wrong. We need think tanks very quickly to evaluate the advantages and disadvantages of any new innovation so that we evaluate our entry point. If it works for us, we take it on that time. If it doesn't, we let it go and we pick another one quickly. Implant is just their way of trying the AI thing, and it is coming. Good people, it is here, and the sooner we start thinking about AI, I know the government has right now a task force on AI. It needs to happen now, because what is AI? This, when you go to the hospital and you open your mouth and we put that thing on you that is able to take photos and sending it at the back. So the question would be, and I'm taking you that level so that you understand where science is headed. Very soon you will have those chips They will come here. You will not have research on them because of the fear that you now have. And therefore, the chips, you won't have control on which one is going into your body. And sometimes there's nothing wrong with being monitored. Monitoring, unless you want to become rogue. But there's nothing wrong with being monitored. So you choose, is it for monitoring where I'm going, which is not good for you because you want to do your things? Or is it for monitoring? If I tell you right now, I want to put a chip on your body that is monitoring the cells and control cell division that can lead to cancer, will you say no? You will say, give it to me. So that it runs in your blood, checks if there is any epitope that shows that there is a cancerous cell somewhere, it has interacted with it, it signals it so that we figure out where it is. Will you still say no to that chips and the implant? So we need to discuss this new innovation for the merits that they bring. Then Africanization, thanks Steve Baraza, it is true. I also strongly believe, and many African scientists believe, that we need to do science and research that fits within our context. And allow me to be a little vague here. Before, there used to be this thing called witchcraft. That if you steal somebody's things, they have a unique way of inflating a pump and your stomach gets inflated. It happened somewhere in Bere. And if there's somebody here from Bere or Embu, it's called Gashamuku or Gaugewa Karanga. That guy was stolen from and by a Makanga. And he said, somebody has taken my money, and he refused. Apparently, I wasn't there myself. He said, get me my bag. He deflated and started pumping. And as he was pumping, the stomach of the Makanga was also inflating. And he returned his money. Now listen to this. Are you aware that is a very serious innovation? So, do you know how much the government is putting in security? If this country, we actually looked at this as serious innovation and we say, in this county of ours, nobody will ever steal any other person. And the source of innovation is this man. And that man, and we package it in different bundles, just like Safaricom. If it is a small chicken, we inflate you for 2 hours. If it is a cow, we inflate you whatever. And it is written very well, we do PR on it. Let me tell you, it is... How different is that thing with somebody who has put a satellite up there for the Safaricom so that you are able to talk to somebody in the US? What's the difference? Where did we go wrong? And that is why the Africanization is very, very key. This country just needs that one person, and we don't have the Department of Police, all of it. We just say Kenya, we as a Kenya, we are going national, you know, and we are going organic with our originality. Our innovation is that the country, and we write a paper and we put an act that no stealing in Kenya. If you steal, we inflate you. The source is coming from the other end. You will not see all these things. It is an innovation, but what have we been told? It is a bad thing. What have we been told right now? Actually, if you are told innovation, somebody is inflating, you run away. Why? So, I agree with you to the extent of how do we go back to our roots. Then we look at what are the basic innovations that we can actually package and give them the modernity that they expect. The disadvantages of GMO are as many as the advantages, depending on where you are looking at. And let me give you an analogy. Vehicles have been causing accidents. Actually, they are killing more than cancer in this country, road accidents. Have we stopped buying vehicles? Why? It is because you are regulating. Water, we are told water is life. And if you take water, it is life. Do you know if you take a lot of water, you get a disease called psychogenic polydipsia? Because of taking the same water that in other quarters we are told it's life. What am I arriving at? I am saying there is nothing that is off the risk. There is everything for regulation. That is why you have guns and you have firearm board that is regulating that. You have vehicles, they cause accident. You have NTSA and road license. You have aeroplanes, you have KA. What I'm saying is GMO must equally be regulated. And that is why NBA is there. The disadvantage that can be there would be, for instance, if I just choose a rogue scientist, I go into the lab and I take a section of the gene that encodes for the venom of a snake and put it into maize. So you eat your maize, you eat snake venom. At that particular point, you don't treat me as a scientist. You treat me as a criminal who is trying to kill people in equal matters. Just like when you have a gun where you are licensed and you have shot somebody, we will still arrest you. So it is a technology All that is important is that we need to regulate it. Technologies have a lifespan. Like right now, the one for taking a gene from this neck to another one, we have gone beyond that and we have not benefited. You know what we are doing right now? It's called genome editing. Genome editing means I'm no longer going to pick the genes from mango to put them in tobacco. No. It is the tobacco itself or the mango itself that I sequence and realize what are the areas that I can tweak to empower it within it. Now that is the latest. It's called genome editing. And it is available, but we are still stuck the other side. Us guys have moved, but we never benefited as a country. Thank
you. Thank you, Professor, for the responses. I said because of trying to manage time, we'll entertain other questions later. But at this particular point, I want to give our VC a chance to interact with the presenter. VC, karibu.
Thank you very much. And I'd like to thank most sincerely the guest speaker. I think my take is that the scientists, you can even see the theme, is science, agriculture, environment, and engineering. They have conveniently left out those people who are able to tell the story. And therefore, they are doing their research, and because the storyteller is not there, so there's nobody to tell the story. So others, the story is left now for those who have no interest in the project. So I think what I'm supporting the issue of multidisciplinary. So let us include the sociologists and the cultural anthropologists to help us also, move this process. Yeah. So when you are doing your, whatever you are saying, genome and so on and so forth, include the storytellers in the process. Number two, I also believe that research, we must be able to drive the research ourselves. The problem in Africa is that we receive a lot of, it's the person who is paying for the research that actually defines the agenda. I don't agree that we don't have researchers who can, research something unique. We have very many researchers. In fact, Africa is the home of the first researchers. The pyramids in Egypt, that is engineering, marvel, isn't it? And who did that? Let nobody lie to you that it was done by Arabic or people from the Middle East or some other strange people. It was done by people like us. Perhaps later they migrated south. Yeah? So nobody has managed to beat that engineering up to now. So that's just my remarks.
Thank you. We have come to the end of question and answer for this particular session. As I've said, if you have any question, we can have the discussion during tea or lunch. Those ones who are online, we'll have the questions written down and we'll see how the presenter will get and respond to the same. I would wish to hand the session back to the DVC, Planning, Research and Extension, to take over from there. Welcome, DVC.
Thank you very much, colleagues. And immediately after this, I think we want to continue. I would like to appeal to you from on the behalf of the organizing committee and the university. We planned for the conference for 2 days, and please, we would like to continue, finish today, and even tomorrow we want to finish early. But we are requesting if we could come early as the program says. We'll do presentations, and during the break we'll have a short time to do some tree planting so that we are doing the same thing that we are doing nationwide. A very short time for tree planting, then we come and wind up with our presentations, because we know some people have traveled from far, and we'll be prepared for this, and we'd like all this conference to come to an end. So that is our appeal, ladies and gentlemen, that please, in your numbers, let's continue the conference, and tomorrow, let's come and finish early. I'm sure the organizers would arrange so that you may finish maybe early by lunchtime, so that you also enjoy part of the holiday, so long as we present all the papers. That is an appeal. So if you know that you are not going to come, hands down. Now, you've not got that innovation. All right. But meanwhile, we just want to appreciate our presenter, and I would like to request the Vice Chancellor just to give something small, just to remember the University of Eldoret. So please, the two of you can come in front. I hope you have the photographer. Thank you very much. Okay, we now invite the director so that you can guide us.
Thank you so much. So when we leave here, we are going to go for a brief
Dr. Bixley Kipsumbai, Professor Ezekiel Kibrop, and Dr. Pascaline Cheruto. All of us are from University of Eldoret, School of Science, Department of Biological Sciences. Xanthomonas is a genus of bacteria known as proteobacteria, and it occurs worldwide, especially it infects crops of economic value like the common beans, cow beans, soya beans. All of them, majority of these vegetables and grain crops are infected by Xanthomonas. In this case, they cause economic losses once they infect the crops, because most of them have been known to cause economic losses of up to 97%. And here in Kenya, some of the losses have been reported. Even in Kalro, there in Makueni, they have reported a loss in cow beef production due to infection by the Xanthomonas pathogens of up to 92%. Also, we take note that the Xanthomonas has got more than one genomic species. What is reported in literature is that they have more than at least 20 genomic species or DNA homology groups. The species have got some significant degree of genetic resemblances, despite all of them being of the same genus. Also, in plant pathology, we concentrate much on the Xanthomonas due to its economic effect, that is, the ability to cause serious plant infections. So, the main objective of our review was to review and look at the genus Xanthomonas classification history, and also look at now the Xanthomonas axonobodies. We narrowed down to Xanthomonas axonobodies. Then we looked at the morphological and biochemical features of Xanthomonas axonobodies. And also, we went deeper and looked at the genetic diversity, pathological traits, and the virulence determinants and virulence genes of now the Xanthomonas axonobodies, Patepa ficnicola. This is the pathogen that causes cow bee bacterial blight disease in cow bees. So during our methodology, this is the method we approached in looking at our review. We did systemic literature analysis, whereby we consulted the databases such as Science Direct, PubMed, Web of Science, Google Scholar, and the Scopus. And we were mainly targeting some keywords in our search, like Xanthomonas axonobodies, cow bee blight disease, and cow bee pathogens. Here, we mainly dealt with one crop, that is the cow bee. Also, we looked at the relevant reports from the government, which have reported the occurrence of Xanthomonas axonobodies pathogen in Kenya and even the other parts of the world. We also looked at the conference proceedings from those who have presented on the same topic. We also searched on other literature to ensure that we had that inclusivity. After the search, we screened our results based on the titles and the abstract to see whether what we had was relevant to our study. Also, after that, we extracted the information and we noted down the key points. So thereafter, we collated our information and prepared one document. So when we come to the history of classification, we grouped the classifications into 4 major ones. The first ones were the classifications that were done in the year 1917 to 1920. And those were the species of Sandomonas that were described then by those who were working on the taxonomy of Sandomonas. And in 1926 to 1983, those were the main Sandomonas species that were described, in addition to what had been described earlier. Although again, during that period, other taxonomic species that had been described earlier were renamed based on new discoveries that were done. Also, between the period 1987 and 1990, those were the species that were described during that period. And in 1995, this is the period when we say that major taxonomic studies of Sandomonas were done. Those are the main species of Sandomonas that were described during that period. And then here we were interested in taxonomic studies of Sandomonas. We grouped into 2. We have the phenotypic methods and we have the genotypic methods. The phenotypic methods are mainly the conventional methods. Then the current techniques that are mainly in use are the genotypic methods. Some of the phenotypic methods are there, including the classical methods, whereby they look at the colony characteristic or morphological, biochemical traits. We have the cell wall compositions. For example, some of the bacteria may be composed of polysaccharides. Also, they looked at that. We have polyamines, cytochromes. And when we come to now, the genotypic methods, these are mainly the molecular methods. They looked at the 16S rDNA. Since this one is the region that is conserved in most bacteria, they looked at the percentage of guanine cytosine in the DNA of the Sandomonas. Also, we have the DNA hybridization. We have the RFLB, that is restriction fragment length polymorphism. We have the RABD, that is a random amplified polymorphic DNA. And then we have the AFLB, that is amplified fragment length polymorphism, and other ones. So these and others are some of the methods that are employed in taxonomic studies of not only the bacteria, even when you are looking at the taxonomic studies of viruses, trees, all those are mainly some of the methods used. And then when we come now, we narrowed it down from the Sandomonas to Sandomonas axonobodies. We've now narrowed to Sandomonas axonobodies species. And in this case, most of these species are plant pathogens. They cause serious diseases of plants. And mainly when it comes to plants now, they are grouped into pathophytes depending on the crop that it infects. Like when it comes to cowbee, for example, the one that has been bolded in black, the Sandomonas axonobodies, that's a pathogen that causes cowbee bacterial blight. It infects cowbees. While the same, if it has got the genetic ability to cause, for example, the other one that has been bolded, it's Sandomonas axonobodies, Patapha faseoli. It infects the legumes. And then, when it comes to morphological traits, mainly, when we look at this photo, it means, when we look at it, most of them have the yellow pigment that gives their colonies the yellow color in nature. Also, some may be creamish in color. Some may be light yellow. Like the first one, the one in A, The one that is a colony of Sandomonas axonobodies, and it's yellow, it has got light yellow colonies. While that of C, the one that has been indicated P first, it's individual colonies of Sandomonas. Well, when we look at the C1, that is rod-shaped cells now of the Sandomonas. They are rod-shaped in nature. And when you come to D, that's now the individual bacterium. That is the Sandomonas bacterium, and individual cell. And then that is a single flagellum in E. That's in aids in movements. And when we come to biochemical features, they are able to, some of them are, they are strict aerobes in that they require oxygen for their survival, and some are catalase positive. All those ones are the characteristics of, biochemical characteristics of Sandomonas axonobodies. And when we come to pathological traits, these are now the symptoms they produce on the susceptible plants. And in this case, mainly the main symptoms are the small chlorotic brownish and water-soaked lesions in plants. I have the image. Those are the symptoms they cause in plants. And in this case, that is a cow bee, which has got typical symptoms of a cow bee bacterial blight. And then when it comes to genetic diversity, when you look at the Sandomonas axonobodies fignicol isolates, genetically they are diverse. And then when you come to virulence determinants of Sandomonas axonobodies, is that we have, those are the genes, virulent genes, which are responsible for causing the diseases in plants. Like, for example, the type 1 secretion system, the bacteria use the type 1 secretion system mainly to adhere to host cells. And when we look at type 2, it's the degradation of host cell wall. Remember, that's a protective cell. And then, in conclusion, we provided, in our review, we provided a comprehensive review of Sandomonas, and we offered valuable insights for policymakers, researchers, and stakeholders involved in plant health management. Also, they start greater public awareness and collaborative efforts that are needed to ensure plant health, to ensure food security and improved livelihoods. And also, we noted that it's important to integrate the pest management system, the IBM techniques, into the management of plant diseases caused by these pathogens. Our recommendations are those ones. The first one is to have a comprehensive strategy to address Sandomonas axonobodies pathogens. Remember, most of them cause serious diseases to plants. Those are the other recommendations. Thank you.
Okay, thank you. Let's we'll allow a few questions. Can you have some questions to the presenter? We have a minute or so for questions. We appear to have no questions, so we'll again appreciate the presenter. So I'm informed that we have another presenter online by the name Naomi, who would wish to, is it Naomi? Not Naomi. By the name Lolimo. Lolimo is online, and I will give a chance to present as we reorganize ourselves maybe after this presentation.
Colleagues, we had to start somewhere. Now we will open another stream, so we for the beginning, we do it this way. Those in physical sciences, cross-cutting, and environment can go to room 1, interested in that. Biological sciences, you can remain here, so that we have 2 teams running concurrently. If we get critical mass, we shall move to a 3rd stream.
Chair, are you able to see my screen? Yes. Okay, thank you so much. My name is Tolimo Festus, and I'm presenting an abstract on allelopathic effect of Achilles toothless, Achilles nubica, and Ivine compressor on the germination of prosopagine flora. Others include Dr. Kenneth and Dr. Bernard Orori, all from Department of Forestry and Wood Science at the University of Eldoret. So under introduction, we know that prosopagine flora is a evergreen, small and fast-growing native tree species that is native to South America, Central America, and the Caribbean region. Due to its early characteristics, including resistance to drought, the species was introduced to other areas in late 70s and early 1980s. In Kenya, the species was first introduced in 1973 at Bamburi for rehabilitation purposes, and later in early 1980s to other parts including Turkana, Baringo, and Tana River, which are all under arid semi-arid. Since introduction, prosopis has become invasive that has caused damage to biodiversity, agricultural system, human being health, and the native plant species. And therefore, we can say that the spread of prosopis has threatened the ecological and economic sustainability into these areas and its inhabitants. So the control and management of many invasive species is key in order to maintain the negative impact below ecological and economic thresholds. Practice pastoralism as a major livelihood strategy. So the three species that is including Facilia totalis, Facilia nubica, and Iphine compressor that were selected in this research are among the dominant and abundant species in Turkana County. And their occurrence has been assumed that because they are still there and they are dominant, they might have allelopathic potential that leads to resistance of the alien weapon by the prosopagule flora. So the leaves and the roots were collected, air dried under shade, grounded to powder for the extraction of crude extracts. And the crude extracts were prepared according to procedure outlined by Omer and et al. 2017, where 100 grams of the leaf and the root powder of different tree species were dissolved separately in 1 liter of distilled water, and they were shaken gently using optical shaker for 24 hours. The solution was then filtered using filter paper and a filtrate kept in the refrigerator as a stock solution. Then this filtrate was diluted to 1%, 2.5, 5%, and 10% concentrations. And prosopagule flora seeds were pretreated with 30% sulfuric acid for 3 minutes to break seed dormancy. So prosopis seeds, 25 of them were spread in their filter paper, placed in a petri dish, where 4 petri dishes were replicated in a germination chamber in a completely randomized design. The seeds in the petri dish were moisturized with 5 ml of each crude extract concentrations, and this was maintained by adding 5 ml of extracts to the petri dish on a daily basis. And distilled water was used in these experiments as a control experiment. Germination count was done at interval of 2 days, and then radical and plumule length were recorded at the end of experiment that took 14 days. Then germination percentage and inhibition percentage was calculated by the formula shown. So under results, as you can see, in the germination percentage, the crude extract inhibited the germination percentage of prosopagule flora seeds. As you can see, the control was 90, and the others are less than 90. So you can also see that increase in the concentration of the crude extract increased the inhibition in the germination percentages. It was also observed that prosopagule seeds coats irrigated with Facilia totalis and Facilia nubica leaf extracts turned dark before germination. And while germination of seed at 10%, extract of Facilia nubica leaves aborted. So seed germination percentage at 10% of Facilia totalis recorded 30 and nubica 23 that were lowest compared to control at 90%. And then the inhibition of the percentage of the germination percentage was between 74 to 21%. On shoot length, the same incident happened where the crude extracts inhibited the shoot length of the prosopis seedlings. And this inhibition was between 8 to 46%. You can also see in the graph that 10% and 5% Facilia nubica extracts and 10% totalis were not measurements were not taken because they died before the experiment period of 14 days. This is the same in the root length, and in the root length, the inhibition was high because inhibition is between 7 to 86%. Same to wet weight, there was also inhibition in the wet weight. Under discussion, we can say that in this research, we focused on the germination because seed germination is a key stage in plant growth, and most of the allelopathy research starts by germination test. Then Facilia totalis, all the three, or I can say the 3 studied native species, had allelopathic effect on the germination of prosopagule flora. As you can see, there was a reduction in all the tested parameters, including germination percentage, shoot and root length, wet weight, and also indicated by the seed coat change in color to dark. And then the change of seed coat can be a reaction by the seeds to the external stress that was exposed by the crude extracts. And then for all, as I indicated, the p-value was less than 0.05, indicating the crude extract affected the germination of prosopagule flora seeds. And then the inhibition percentage of the crude extract differ depending on the plant parts, and the crude extracts concentration. And in generally, an increase in crude extract concentration led to increase in inhibition effect on prosogyl flora in this study, as also proven by the previous study done on allelopathy. To conclude, I can say that the native tree species studied have allelopathic effect on the germination of prosogyl flora, therefore can be promoted to suppress infection of prosogyl flora that is invasive. This study has also shed light on the utilization of the native species in the control and management of invasive prosogyl flora. It is therefore recommended that study on allelopathy effect be done on the growth of prosopis and also field assessment. And then further study needs to be done in other dominant native tree species in areas where prosopis is embedded in order to identify more native species that can be used as biological control. Thank you so much.
Thank you for the presentation. We appreciate your presentation. I'll allow 2 or 3 questions. Any questions? Okay, there's one question over there.
Thank you. My name is Max Longura, a PhD student. Thank you, Lolimo Festus. Mine is just an observation, especially to Lelimo and other presenters who are about here too. When you do your work, we want to believe this is your personal work, own it, so that when you come to present before us, the information on the screen, it is for us to read. We expect you to have mastered your work, and in particular, these are published papers or so. So just mention in passive and let the rest of the information for us to digest, so that when it comes to question session, it will be easier. Thank you. Because if you check on the aspect of introduction, there was a lot of reading, and on results, he went through it very quickly. Thank you.
Okay, so that's just a comment. I hope it has been taken in. Any other comment, any other question? Yes, Dr. Kitilit.
Prosopis juliflora has been known to be a very invasive plant in the areas where it grows, and it has been known to suppress all other plants very quickly. I don't know how it works so that other plants now, the researcher is reporting that other plants can be used to suppress its growth in the environment. I wish there was some pictorial or something to be able to show that.
So the presenter, can you take up that question?
Thank you, Prof. It is about, I'm sorry I didn't put some pictures to prove my argument, but my work is fully based on homeland security theory in allelopathy study that indicate that although alien invasive species affects other native species, the native species in the area, at times the native species might be resistant to the alien one. And I did my literature review, and I've identified also some species are still occurring together with prosopis, which is against what we expected, that all species should have died, since prosopial flora is more invasive. That I can use the term, will kill all the other species and remain to be the dominant one. But in Turkana, the research done in Turkana have indicated that the 3 species that I've taken for my research, among others, are still dominant in Turkana County. Thank you, Prof.
Okay, thank you. Thank you for responding. Any other? I think if there is none, we can appreciate Lolimo. I'll invite the next presenter, who is Dr. Kitilit. I think he has some talk on aflatoxin, blah, blah. So welcome. And then after Kitilit, we'll go online and then come back to our program.
Yes, good afternoon, ladies and gentlemen. My presentation is on aflatoxin and the rabbit. Yeah, and there it is. This is, I studied the association between aflatoxin and aflatoxin on one side, the effects it had on the hypothalamus, pituitary gland, and gonadal tissues, that is the ovary, and other organs, as opposed to the hormone levels in the diet of the rabbit. And my name is Kitilit Jackson from the Department of Animal Science. On the introduction, aflatoxin, maybe I should have said that, is one of the mycotoxins. There's several mycotoxins that appear in life. They are everywhere. There are mycotoxins in water, in the air, everywhere, and there are others which are, cannot survive in our tropical environment, so they survive in the temperate, but there are others which are also local to the tropical environment. And aflatoxin is such a one which appears in the tropical environment. And work that had been done by Kangede and Lang in the urban areas in the country showed that there were quite high levels of aflatoxin among the feed manufacturers and the feed handlers all over the country. And they found that some feed samples contained up to 81% in Nakuru and Nairobi. And we thought there might be some effects on the fertility of the rabbit, which is one of the animals that have been domesticated and are fed intensively. And fertility is one of the key factors that determines production in rabbits, because a kitten has to be born and grown for slaughter. So therefore, the more the kittens that are born healthy and are able to grow well, the better for the meat. Recent studies, of course, have shown that there have been interferences of aflatoxin on reproduction at various stages, and many studies showed that, studied the effects, particularly on male and particularly on the spermatozoa, but little had been done on the female. And investigation of aflatoxin exposure in utero showed that even growth and development of the fetus has been interfered with in the past. Among the female rabbit, aflatoxin has been known to diminish fertility of the oocyte and disrupting their maturation, and even by way of epigenetic modification and oxidative stresses, and that is work reported by Liu et al. In 2015. Therefore, the objective of this work was to determine the association between aflatoxin content, the level of histological damage or of the organs that play a role in reproduction, and the hormone levels in the rabbits. In the materials and methods, we prepared aflatoxin in a laboratory and incorporated in the feed at various levels as that. Diet 1, of course, was to serve as the control, and we ensured it was free from aflatoxin. Then treatment 2, 100 parts per billion. Treatment 3, 200 parts per billion, sorry, that should be 3. And then treatment 4, 400 parts per billion. And we fed to rabbits, 16 rabbits for 45 days, during which period we collected blood every 4 days and tested for the hormones, luteinizing hormone, follicle stimulating hormone, and estrogen. One rabbit at the end of the study was taken to the laboratory for histological study, and the organs were removed and examined under a microscope after the organs were harvested. The data was subjected to various statistical methods and particularly the methods of relationship between the effects and the levels of aflatoxin and the level of the hormone. Now, the changes were scored. The histological changes were scored, and the issues of concern were inflammation, cell necrosis, vascular degeneration of the cells, cell infiltration, and so forth. And we gave them codes. 0 was the organs that were not affected, and 3 were the most severely affected. And the measurements were, the coding, the measurements and the codes were used for analysis using Kedel tau rank correlation method, and it gave us some results. So the results I observed in the work is that there were significant relationships and interactions among the various components, and of great interest was the first one, which is the aflatoxin level. And aflatoxin level related quite negatively with the liver and the ovary. And these were not quite high at 0.6. That means we are at the coefficient of determination. It is average, but the effects were significant. Again, of interest was particularly FSH and LH was significant in relationship with aflatoxin. And we thought that it represented an indication in which in a situation where there is a lot of hormone sensitivity in the animal, for it to be in a reproductive state or to be fertile, these levels and that relationship indicated that there would be most likely a problem. Hormone levels significantly correlated negatively with aflatoxin. The luteinizing hormone was particularly negative. And you know this is a very important hormone in reproduction in that the level has to reach its threshold amounts for it to be able to cause reproduction in the rabbit. And therefore, any indication, anything that indicates that it went down or it negatively affected it would easily also affect reproduction. In conclusion, therefore, there was a negative association between aflatoxin and the hormones, and this was very important. There were very minor relationships with the other organs, but I couldn't include it in my conclusion because it was solo that it might not be of any serious consideration maybe in reproduction. Minor negative correlations between histological damage and the hormones were observed. And again, there were quite a number of issues that we thought of here, particularly some of the hormones, like when it related negatively with, if you have FSH and related negatively with the hypothalamus, then you know that hormone doesn't come from there. I think the best hormone I should have analyzed here was GnRH, and then it would have related properly with the hypothalamus. My recommendation, therefore, is that diets of rabbits must not contain aflatoxin above the allowable levels of 50 parts per million. This is the recommended levels. But other workers have said even at this level, it still affects various components. Thank you very much. I will end there.
We'll entertain questions. Any questions, any comments? So any question, any comment? Okay. Dr. Pascaline.
Okay, thank you, Dr. for that presentation. I'm just want to know that from your work.
Yes.
When you are doing this, at least you did some of the literature review to find out the correlation between the aflatoxin and the fertility. Is there any relationship in that case with the human fertility of that concern? You remember even in the GMO, when you talk of GMO affecting fertility, it is like, what? So that is also my concern as we consume food, and we know aflatoxins is of importance to man.
Okay, second region, there's a lot. And being all biological, aflatoxin would affect both all the consumers that consume it in terms of fertility. There is no direct study on human effects, but it has been done for human, it has been done for the growth of children, mothers that consumed aflatoxin infected feeds, the sizes of their babies, and so forth. and it has been found that it affects. And as I said, work that has been done on other animals have shown that in utero there is effects. The formation of the fetus is interfered with, and then of course the size of the fetus once it is born. So the indications are that they have an effect. Yes.
Thank you.
Yeah.
Maybe to second on that.
Yes, please.
Why did you decide to use the rabbit and not the mice? As in most cases in science, we prefer mice than rabbit.
Yes. I don't have there was no major reason why I did that, but I read in literature somewhere that the rabbit, the genetic reactions of a rabbit can easily be more or less similar to those of human. So it was based on that I thought maybe I can go that direction. Yeah, but I could have used the mice and so forth. Yeah. Thank you.
I have another question. Yeah, just a short one. I also wanted to ask the same question that Dr. Pascaline is asking, because we have rationales behind the test you're doing and the model organism that you want to use.
Please, loud.
All right. It's a good research, and we can use that to model human diseases. We can use...
I can't say really I was modeling.
All right.
I was just doing an animal study.
Okay.
Yes.
So it ends after the study on animals, but do you want to use the
Of
results?
course, somebody else can pick it. I'm an animal scientist. I am not a human scientist. Of course, the other scientists, human scientists, are doing their work elsewhere.
All right.
Yeah.
That's okay, and I wanted to support that there is a lot of conservation between rabbits and other animals and human beings. There is a close link. I'm using frogs myself to do research. I'm using Xenopus lavis, species of frogs, which have been used globally to model human diseases and also testing of drugs and other things. So I agree to the answer you gave to Dr. Pascaline. Thank you.
Thank you. Thank you, Bwana Chairman. I think I stop there. Oh, there's one.
Okay, thank you, Taksari, for the presentation. That's been an enlightening one. But during your presentation, I've been asking myself some few questions. One, is the reduction in the fertility rates of the rats directly as a result of direct effect by the aflatoxins, or as a result of the negative effects that the aflatoxins have in the signaling pathways of the release of these hormones, or as a result of direct negative effect on the organs responsible for the production of the hormones? That's one question.
Yeah.
And then the other question is relating on the, during your presentation you said that you prepared the aflatoxin. Yes. So you prepared the aflatoxin itself, or you fed the rats with the feed contaminated with the aflatoxins?
Yeah. Let me start with the last one. We prepared the aflatoxin and then incorporated it with into the diets. Yeah. Then the other question is on, remind me again.
The other question.
First one.
Let me still proceed with the second question.
Yes, the receptors.
Yes.
Yeah, I didn't quite go in to separate the effects of aflatoxin, whether it went to the receptors, whether it went to the other communication pathways, the signal pathways, and so forth. I didn't go in that far. But literature is awash that there are effects on receptors, and there is a possibility it could also affect the signal pathways, the communication pathways within the system. So, but I didn't go into partitioning that much. I only went into the main organs. Yeah, that I reported on.
Okay, thank you. And the last one.
Yeah.
During your pathological studies, did you maybe examine the rat meat, not the rat, the rabbit meat?
Rabbit meat.
Yes, to check if it contained the aflatoxins.
No, I didn't.
Okay, thank you.
Yeah, thank you. But I didn't, but I had comparative, of course. I had the control rabbits to help me in studying the rabbits that had interventions of aflatoxin. So I was able to deal with the organs directly without necessarily checking the amounts in the animal's body. Because already in the diet of control, we had controlled the amount of aflatoxin to almost zero. Thank you. Thank you, Bwana Chairman.
Okay, I'm just wondering about the food thing, considering rabbits can be food to pets.
To pets?
Yes, apart from being human. So I was wondering if the aflatoxin toxin travels the food chain. So you answered part of it, but I'm wondering, let's say they've fed on aflatoxin and then like a dog or something eats that rabbit.
Yeah.
Does the toxin travel along the food chain?
Yeah, to the dog. It still does. It still does. Of course, once the animal takes it, through the metabolic processes, they are converted into various other components which are not directly maybe the particular toxin the way it was. It is converted into another component. And therefore, any animal that will consume that animal, like now, the animal that will consume the rabbit, like the dog, will still pick the aflatoxin, but not in the same compound as it was. And maybe in the same vein, it will not be as effective in causing maybe pathological damage and so forth in the next chain of the animal. Thank you.
I think we'll stop there for this session. I'm informed we have a presenter online. Do you have somebody online? Somebody's online, eh? Somebody, Mercy. Yes, Anu Asike. Yes, Anu Asike. So we'll have Anu Asike presenting, and then we come back to our program. This session was supposed to be for the online session, so we are trying to mix online, physical. Yes.
He asked me whether in terms of gender, there is a difference in terms of effect.
Yeah. Maybe as we are waiting for the online presenter to connect, you can tell us something about gender. It was related to what I think Dr. Pascaline asked, but you didn't answer fully. You can comment as the online presenter.
Literature says children and women are more disposed to aflatoxin more than men. The reason is obvious. Women are the people who handle food from the time of preparation from the stores to the time it is utilized as food, and therefore they are exposed. When you go to a place like Ipchoge, you go to town and these farmers have anchored their maize. They anchor under very unmanaged conditions. The children and the women who have no food will come collecting the grains, and at the process they collect aflatoxin, and they are therefore more exposed, predisposed than the men. That's the reason. Thank you.
So I'm informed, Anne, you can continue. I know the topic is non-biology, which has facility for online presentation. So I think we'll just try to listen to her and ask questions. So Anna, are you okay? Can you proceed?
Yes, Chair.
Okay, thank you.
Thank you very much. My name is Anne Wasike from the School of Education, the Department of Educational Psychology. My topic is psychosocial intervention strategies to manage stress among student mothers in public secondary schools in Kibilili Sub-County, Bungoma County, Kenya. In this presentation, I am with Dr. Oseko and Professor Simiyu, all from the University of Eldoret. Welcome. As a way of introduction, students who are in public secondary schools are presumed to be minors and also single, and therefore have no accompanying children for that matter. But from the literature that I have had, very many girls in public secondary schools have fallen victim of early pregnancy. And these early pregnancies have served as a threat to the girls. It's very good and important to look at the psychosocial intervention strategies. When a girl finds herself in this situation, usually or normally there will be increased anxiety. They may suffer frustration, and to some extent they may drift into depression. Why so? Because maybe there is lack of these psychosocial strategies to help them remain in school and work and be able to achieve academically. In many situations, there is lack of emotional support, and this therefore leads to alienation of the girls from life. When many times, even when they go back to school, there are no strategies put in place to help them settle down psychologically, physically, even to some extent economically. And so you find that their performance is not as good as is expected. Therefore, this research was intended to assess the rate of intervention strategies, particularly in Kimilili Sub-County of Bungoma County, Kenya. The objective of this research or studies was to assess the rate of stress among the student mothers, particularly in the public secondary schools. How are they doing in regard to their school life as students? So we also realized that at that particular point, many a times the teachers and the environmental situation does not allow them to fit back in school and therefore continue to achieve their academic intentions. The methodology that was used in this study was actually the cross-sectional design, which meant that we obtained both qualitative and quantitative data, which was later on analyzed to give us the results that I'm about to give in a few minutes. The population that was indeed targeted was 381 students in public secondary schools from both pure girls schools and those who are in mixed secondary schools. And then we also targeted 19 guidance and counseling teachers and another 19 school chaplains. These were the respondents whom we used to obtain the data that I will analyze or talk about in a few minutes. Before we went back to the field, we went to obtain the large scale data. I did piloting in order to determine whether the instruments are able to get the desired data and information from the respondents. The hypothesis for the study was that there is no effect of rate of stress on student mothers in public secondary schools in Kimilili sub-county of Bungoma County, Kenya. Also, with that, then I can look at the results. What results were obtained? Indeed, the results came out to nullify the hypothesis, and therefore it was discovered that there is significant effect on rate of stress among student mothers in public secondary schools in Kimilili sub-county of Bungoma County. We went on to do the discussions and discovered that indeed there is depression among these students, those who are in school, and to some extent, even when they're out of school, they suffer depression because they find themselves at crossroads, torn between the academic work and motherhood for that matter. So they have quite sufficient challenges as far as they're concerned, they're being in school and so forth. And more so, to a great extent, the student mothers did not get this support that I really wanted to establish while they're in school because of the situation in which they found themselves. Remember, the teenage or teenage pregnancies, to them, usually causes it destabilizes them because many a times it is unwanted, but to some extent they find themselves in the situation, and so they sometimes drift into depression both in school and out of school. And ideally that means that it causes lots of psychological effect on the girls because they have dual roles to perform, and yet they, to some extent, in some cases, they are single-handed. Therefore, early pregnancies do affect their school completion a great deal, particularly when you look at the good amount of literature indicates that in many communities, girls who got pregnant while they are in school were declared or handed into marriage rather than helping them to go back to school to enable them achieve what they can achieve as far as education is concerned. So you find to a great extent there is a cyclical, of occurrence of even poverty, because in cases when a girl gets pregnant and comes from a humble background, that will mark the end of her schooling, and therefore it will end up not changing the economic situation of the family, and that is rather unfortunate. And besides that, these early pregnancies has also led to lots of effects and has caused what we call a gender divide between the boys and girls, because in the literature again, it was discovered that some girls drop out of school even when the boy in question was also a student. The boy will continue with their studies and the girl becomes a victim of circumstances. So then it creates lots of gender divide in that particular case. Literature also has it that, or previous studies have indicated that Kenya, for example, among the Kuria, they quickly married off the girl as soon as they discovered that she was pregnant in order to gain or earn dowry for the family. So it really is an issue that I thought was worth investigating. Besides that, these early pregnancies have gone ahead to affect the school re-entry policy. This the return to school policy indeed is in place, is available even here in Kenya from the year 2006, according to data that I came across. But again, many parents, of course, plus the children, are not aware of this particular policy, and therefore the girl ends up, terminating the education so that they are not able to continue with education per se. And then, of course, the issue really escalated with the COVID-19 pandemic, where indeed it was all over the news, in the literature, and every other place that there's a very high rate of teenage pregnancies, and yet the government declared that no girl should be left at home. But yet, many of them remained behind and did not go back to school. Those who went to school were not, made comfortable to enable them achieve academically. Also, therefore, it really thought it is an issue to investigate. And then, of course, this lack of knowledge on the psychosocial intervention strategies has indeed caused the girls lots of challenges, and how we wish that we could see what can be done in regard to the same, so that the girls can have these psychosocial intervention strategies to help them to settle back in school. In the discussion, the prevalence of the girls who said they have no psychosocial support was about 56%, and then who didn't have psychosocial support was 56, and those who felt they had were just a number of 44%. Also, in the discussion, I would indicate here that the 73% of the girls who were interviewed were able to carry their pregnancy up to delivery, whereas 27% may have lost their pregnancy along the way. And again, this is a matter for another day. The third discussion point that I've mentioned here is that the age of the girls who were respondents in this research included that 6% of the girls were only 13 years old, 9% were 14 years old, 15% were 15 years old, and the 16-year-old girls took 34%, and then 27% were 17-year-olds, 5% were 18-year-olds, and 19 and beyond were only 4%. So indeed, this confirms the fact that these girls are indeed fall victim from the age of 15 up to the age of 17. And that is indeed the secondary school going age. In conclusion, the study indicates that stress is positively related to performance, that these particular girls who are victims of early pregnancy are, their performance is greatly affected because of lack of this psychosocial support that we imagine should really accompany them where they are. The second conclusion is that there's a high relationship between effective counseling and the level of stress and stress management among the learners. In a short while, we shall be concluding that guidance and counseling is an element that should indeed be escalated in the school system, particularly for students and girls who find themselves in such a situation, in order to help them cope with the circumstances and the situation in which they are. Thirdly, the research concluded that there is positive relationship between social support and stress management, that in case these girls get the opportunity to gain social support, then they are likely to achieve even academically. If not, or otherwise, stress is really an issue in regard to them. With that, Chair, I rest my case. Thank you.
Okay, thank you. Let's appreciate the speaker, Anne Wasike. Do you have some questions, comments on that presentation? Yes, I can see three from the audience. So I think we'll start with Dr. Achonyo, then we go to Maxwell, and then Ezra, you'll wrap it up. So just go straight to the point.
Thank you, Chair. Thank you very much, Chair. I just want to point out that the presentation was reversed.
Okay.
Discussion was done and then results presented last. Okay. Results should have been presented and then discussion follow. Because we saw the tables and whatever, the figures coming and conclusions, which should have been up there. Okay. But I want to ask on the conclusions. We didn't see this conclusion material being presented or investigated. Even when she was presenting about the hypothesis, I don't know whether she said accepted or rejected, she had not shown us that these were the results that were directly related to this study. So the results do not match the conclusions and are not presented at the place where the hypothesis is being rejected.
Okay, I think, Anne, you have taken the comment, so you'll respond to that. About the order of presentation, I think that one, you don't need to respond. That's supposed to be the order. Maybe there was some problem with technology, but normally you present the results and then you discuss. But there's something which Dr. Achonyo asked about the conclusion. You might need to respond to that.
With what Prof.
We understand that reproductive health starts way from class 6. And we expect that maybe by the time they're in Form 3, they should be having some knowledge. During your study, did you do any assessment of the same? The relationship between the reproductive health and the number of, I can call, accidental or planned pregnancies. And then the other one is the issue of the mental capacity of the pregnant teen mothers and the
recording in progress
and the number of pregnancies. Let's say, for example, did you maybe record the number of pregnant teens in national schools to those ones from county and extra county schools? Where are we having the largest number of pregnant teen mothers? From the county and extra county schools or from the national schools? That will inform us the mental judgment and the issue of pregnancy. Those were my concerns. Thank you.
Okay, thank you, Ezra, for those comments and questions. Anna, are you online?
Yes, Chair.
Are you able to pick up to take up the questions?
Yes, Chair.
Okay,
Though
thank you.
I got offline a little, I didn't get the last question from the last person.
Just answer what.
Possibly, let me respond to the first questions that came earlier. For the case of the order of the report of the results and the recommendations, thank you for the input and your support on that. I'll take it up. Then on the bit of having supportive results, statistically, I'll say yes, because I did not capture them in this brief presentation, but in the The main document, they have been presented in terms of tables and percentages. And so I'll say, yes, I have the results which support these particular findings. And then for whether national schools and so forth, I did a purposive sampling, though unfortunately we did not have a national school in that sub-county. So I worked strictly with the pure girls schools and then girls in mixed secondary schools. I hope I have answered you adequately. Thank you, Chair.
Ezra, all your questions answered? Which is the other one? Okay. Which one has she not answered? You can ask me, then I just ask, and then we. Okay, there was a question on the knowledge of reproduction, health, and pregnancy in high schools. Hello, Anne? Hello? It's like
yes, I can hear you.
Oh, there was, I think, a member wanted to know to what extent this knowledge of reproductive health and pregnancy has been sensitized to, I think, schools.
Thank you. A good question that is. Indeed, according to the data obtained, it was realized that not many schools and not many student mothers have this knowledge, and indeed that was part of my recommendation as is indicated at the end of the work. Not many have this, and more so, there are lots of questions from religious bodies regarding sexuality and sexuality education in schools. So it looks like not many girls, and to some extent parents, have this information. So it is quite an issue that can be researched on at another point in time. Thank you.
Okay, thank you. I think I appreciate the presenter once more. Okay. So we can maybe do one more presentation as they are organizing, I think, for lunch the other side. My list is a bit distorted. Something climate change. Let me see. Yes? Yeah, okay, thank you. I'm told we have one more presenter online. We can allow the online person to present. Those who are here physically, I think we have no problem. Let's allow Mercy or she's physically. Okay. Okay. So that's Mercy Kavila. Okay, I thought she was online. So Mercy, you can come and present from this side. Any other person whom I had called and is still okay? So no problem. We'll still have another session after lunch. But Mercy, you can.
Good afternoon. My name is Mercy Kavila, and I am from the Department of Mathematics and Computer Science. My work here is very simple. I want to talk about physical inactivity among the University of Eldoret staff. So, to begin, why we are talking about what is physical activity, it's any activity that involves the muscle movement, and in the process, energy expenditure is used. And therefore, when we talk of physical inactivity, it is the failure to meet the recommended PA levels. PA is the physical activity. These recommended PA levels are given by the WHO. So, physical inactivity is a risk factor, or we can call it a modifiable risk factor to non-communicable diseases. And these non-communicable diseases are the diseases that nowadays we call them lifestyle diseases, like cancer, type 2 diabetes, we have cardiorespiratory diseases. And when we talk physical inactivity and the burden that NCDs are bringing to us when economically and health-wise, the more burden is experienced in low-income countries, in which Kenya lies into. So, when you look at the projections that we have, we see that about 52 million lives will be lost to non-communicable diseases by the time 2030 is here with us. And we have seen that WHO has done a very good work in coming up with strategies, coming up with innovations, in measuring, monitoring, and evaluation of physical activity. We have also seen a reduction in occupational PA, whereby we have switched to more technology and somehow shifted from what we used to do, like active transport, like working manually, and this has also brought to sedentarism. Sedentarism is having long sitting hours, and that has also reduced the physical activity in our lives. So, since we know that physical activity is becoming a leading cause of non-communicable diseases, and reduction in occupational physical activity has also cropped in and has increased the risk of us developing lifestyle diseases, we have seen that in 2022, Kenya recorded 120,000 deaths due to these lifestyle diseases. And there's a probability that of 21% that someone with non-communicable diseases will die prematurely. And in our institution of learning, there's been noted to have a high reduction of occupational physical activity, which has increased our risk of developing these non-communicable diseases. So why this study is because we'll be able to understand the contributors and the consequences of physical inactivity in institution of higher learning. Also, we shall get to know the link between physical inactivity and how it is affecting our organizational performance, bringing up poor service delivery, and staff health is affected in a way. It can also form a baseline for other institutions of higher learning on physical inactivity, non-communicable diseases, and mitigation measures. So the objective of the survey was to investigate the prevalence of physical inactivity among the staff of the University of Eldoret, and also to explore the associated contributing factors that can limit a staff from consistently achieving the recommended PA levels. This survey was a descriptive survey that was done at the University of Eldoret. It targeted both the teaching and non-teaching staff, and in the process, 321 staff members consented to the study. And we used SPSS for data entry and R analysis. The presentation will be in descriptive analysis and also some inferential statistics. So when we come to the results and discussion, as you can see that overall physical inactivity in the University of Eldoret is 36%, which when you compare it to the global physical inactivity prevalence is 25%, which is 25%, we are a bit higher than what the global prevalence is at. This shows that there is high prevalence of inactivity among our staff. So we went ahead and did an internal evaluation to see whether the teaching staff or the non-teaching, to compare the two, who are more physically inactive than the others, and we could able to derive that the non-teaching, as far as they recorded that they have perfect, good, and averages were high. On the lower side of unfit, the same non-teaching recorded high values. This can be attributed to the fact that most of the non-teaching staff spend their time seated in the offices, unlike the teaching staff whereby they have some movements from one class to another, from one department to another. So that can somehow attribute to the physical levels of teaching staff being a bit higher than the non-teaching staff. So through multivariate logistic regression, this was to look for the correlation between the factors that we were trying to measure that can affect the physical activity in most of the people. We did multivariate logistic regression using R, and things that we thought like gender, age, job category, and education levels, we found that they were neither a risk factor for physical inactivity among staff, but BMI, wealth index, and knowledge of the respondents was considered to be one of the things that can influence whether you participate consistently in physical activity or not. We used a p-value of 0.1. So this is the results from R. When you look at it, and where there is asterisk, it shows that it is in the PL level 0.1, above 0.1. So you can look at the income. People with lower income, like below 40%, 40,000, and this can be explained that most of the people think that if you want to be physically active, you have to go to gyms, you have to attend, and most likely is that these people who have income that is lower than 40,000 may not be able very much to pay the monthly subscription to these gyms and everything. And then you can see that BMI of category 4, those are the people who are above the normal weight. Most of the time we call them obese, but above the weight, again, it was one of the things that was affecting the physical activity. And then exercise plan. We wanted to know whether people, our staff have an exercise plan whereby they know which physical activities they are engaged to and when to work out. And those who responded that they didn't have a physical activity plan were found to be likely fall under physical inactive group. So in conclusion, we can say that there is high prevalence of physical inactivity among the staff of the university. We can also see that from our internal evaluation of fitness by cadre, non-teaching staff are likely to be physically inactive compared to the teaching staff. And we can also conclude that demographic factors are not likely to contribute to physical inactivity. But if we look at the BMI, people who are a bit overweight, and the wealth index, and also people who have physical literacy are likely to be physically, people who lack physical literacy are likely to be physically inactive. So as we conclude, we can recommend that through public awareness, through public education, we can educate our staff about physical inactivity, about the link between physical inactivity and non-communicable diseases, and the personal effort to physical activity, because health is something that it's a personal. It is at personal level. As much as the university can try their level best to make people physically active, you have to make it your personal effort to be healthy. And that's why we are saying through sensitization, we can sensitize our staff on the need for physical activity in their lives. And also that there can be more research on our institution of learning and the link between now physical activity, physical inactivity, non-communicable diseases, and how it is going to affect our productivity in the institution of higher learning. Thank you.
Okay, so let's appreciate Mercy once more. I want to vary the order of presentation a bit so that we manage time. I don't know how many presenters are around. Okay. So what we'll do, you'll reserve your comments and questions that you wanted to ask Mercy, and then I'll allow the other 4 presenters to present, and then the questions will come at the end. And we'll be very strict on time. We want to finish by 4. So we'll give you 10 minutes. So please, those ones coming to present, strictly 10 minutes each. 10 minutes. So what's the order? What's your name? Oh, Naomi can come, I think. So Naomi, strictly 10 minutes. Go straight into the point, introduce, and then give us your results. And then the other person can also be ready. So strictly 10 minutes, and then we'll ask all the questions together, not your questions. Even for Mercy. Mercy, don't leave. We'll ask the questions at the end. Thank you.
My name is Naomi Awino Okumu. I'm presenting under the sub-theme Environmental Health and Livelihoods. The topic is Datura stramonium, evaluation of its potential as a biocide for mosquito causing malaria. This is a review, and as we were doing the review, the main question we were asking ourselves was, how effective are extracts of Datura stramonium for control of mosquitoes that cause malaria? Now, there's a problem here with mosquitoes, and they cause many human diseases. One of the most important diseases caused by mosquitoes is malaria, and from WHO 2023, quite a number are being affected by mosquitoes globally. We have about 5 million people affected. 2 million reported cases in the hospitals, and 600,000 annual deaths. Amongst this population, children are mostly affected, and unfortunately in our country, 70% of the population is at risk. The other problem is climate change that has doubled or times 15 caused the risk of malaria, coupled with the humanitarian crisis due to flooding like has been going on. And then there are also invasive species of mosquito causing malarias that have invaded our country. Economically, a lot of money has been spent to control mosquitoes that cause malaria. And so far globally, $12 billion have been spent on control. And in Africa, 4.1 billion, while in Kenya, 33.5 million Kenyan shillings have been used to control mosquitoes. And so you can imagine how much money that has been spent because of the mosquito issue. Another problem with the mosquitoes that are causing malaria is because they have to be controlled, this has resulted in a loss of 1.3% of the GDP, and bringing a socioeconomic threats. We are getting broke because we have to fight malaria. Another problem, which is now of our interest in this review, is the vectors and the parasites are developing resistance to the chemicals that are being treated to treat them, while the parasite is developing resistance to the medication that is being issued to people. This is by Ochomo et al., 2023, and it's interesting that 78 countries have confirmed that mosquitoes in those countries are resistant to the control. So this means the effective control procedures that are in place have been slowed down. As much as there's a try to control malaria-causing mosquitoes and control malaria, prompt diagnosis, treatment, prevention in the form of prophylaxis and vector control has been put in place. Remember, vector control is our interest in this review. But then, the vector control tools in place have got challenges, like internal residual spraying can only control indoor biting mosquitoes. Insecticide-treated nets, people have attitude about them, they wear out, and they can also only control indoor biting mosquitoes. Larval source management also has a challenge because of the continuous human activities that create new sources. There are other tools that have been used, but they are equally expensive with their own challenges. Generally, according to WHO, there are several methods of controlling the vector, which is our interest. The chemical pesticides, which I've said, have gained, the vector has gained resistant to it. There is biological control with the enemies and the bacteria and viruses. There is insect sterile techniques where male mosquitoes are taken through radiation and then they produce infertile sperms, infertile cells for reproduction. Then there are cultural practices and environmental engineering, and finally, botanicals, which is our point of interest here, because even though they are slow in acting and require periodic repetition as application, they are very, very eco-friendly. Why are we interested in this review? We are trying to see if the issue of resistance can be sorted by seeking to find alternative eco-friendly control methods, which can be used in insecticide, integrated vector management approach. Now, because of this, bioinsecticides is a promising tool, and botanicals are bioinsecticides. They have multiple modes of actions that makes them not really able to be resisted by the theme of this conference. Now, a few of these botanicals are registered, The widely researched ones as a director indica and pyrethrins. And therefore, there's a problem because there is not enough information on botanicals that should be considered for registration. To get information about Datura, we used web-based literature search, and we took the period 2010, between 2010 and 2020, and we searched in Google Scholar, ResearchGate, PubMed, and Elsevier. From our research results, Datura stramonium is very poisonous and medicinal at the same time. It is a flowering plant of the genus Solanaceae. It grows everywhere in almost all environmental conditions or climatic conditions, and the entire plant, interestingly, contains phytochemicals such as alkaloids, terpenes, saponins, flavonoids, and tannins, sugars, and organic compounds, which are very insecticidal, according to Alsnafi 2017. It is of our interest because it is locally available as an invasive noxious weed in Kenya. The results of this review. Most of the studies on Datura stramonium focused on the pharmacological properties. This can be confirmed with a study done by Sharma et al. 2021. Datura stramonium has phytochemicals and compounds which are antibacterial, insecticidal, and antimalarial. A number of studies have confirmed this. Interestingly, most studies on this were done in India and Pakistan. Not so much has been done in our nation. Most of the insecticidal potential studies done used larvae of unknown resistance status. They just picked on a species and then they used the larvae for the experiments. Then the antimicrobial tests focused on bacteria that affect human health and not those that mediate resistance. It is important we understand that currently there is a type of resistance in insects that is mediated by microbiome or microbes that are living either in the insect or on this body surface. What is the implication of this review? There are gaps that we came up with from this review, that effectiveness of resistance mosquitoes and insects are not well understood in terms of Datura stramonium extracts. People don't know, there's not enough information to justify if Datura stramonium is effective on mosquitoes that are resistant. Reports did not dictate or say whether they were resistant or not. Another gap is inhibitory effects of the microbiomes community of the mosquitoes are not fully well explored. Of the mosquitoes that were tested, it is not clear if the microbiomes were equally affected now that they are microbiome-mediated resistance. It is also not clear that Datura stramonium can effectively work synergistically with the existing chemical pesticide. If it is mixed, can it work? Some compounds of Datura stramonium have been tested, but some have not yet been tested to prove if they can be effective on resistant mosquito species. The big question this review is trying to ask is, how can phytochemicals and compounds of Datura stramonium be of importance in controlling microbe-mediated resistant vectors and pests? Thank you. God bless you.
Okay, let's appreciate Naomi once more. Okay, so reserve your questions and comments. We'll have questions and comments coming in after the presentation. The next presentation. So next. So the other presenters who will be coming next, I will request that you just sit in the second row so that we can easily identify you. I think there are two more. Are you ready? So 10 minutes, and then we'll take the questions together.
Time in the discovery of new drugs. Ximenia americana belongs to Olasai family. It's a bush-forming shrub around 2.7 meters high. And it is used for various purposes like fruits, medicine, construction, and additional medicinal uses of Ximenia americana in Eastern Africa, and also to establish and document to establish the documented pharmacological activities of this plant, and also to establish their phytochemical profiles, also documented from literature. The method employed include literature search. So the literature search strategy involved search on traditional uses, phytochemistry, and pharmacological activities of Ximenia americana. This was done from September 2023 to March 2024 using Google Scholar and PubMed. This study covered a period from 2010 to 2024. Key terms used in the search were ethnobotanical surveys of Ximenia americana, traditional uses of Ximenia americana, pharmacological activities, phytochemicals in Ximenia americana, and also using English nicknames of the plants like wild plum and sandalwood. The inclusion criteria involved all published ethnobotanical reports reporting on Ximenia americana were from 2010 to 2024 were included. Information which did not report, or ethnobotanical surveys which did not report Ximenia americana were excluded. Also, review articles were excluded. Only original research articles were included in this study. For screening, this was done by screening the titles and the abstracts of identified journals. Those ones were downloaded and used for purposes of compiling data. And the data analysis was done by the information from the traditional uses, the plant parts. All those were entered into Microsoft Office Excel format, then exported to SPSS. Statistics used included percentages and frequencies were used to analyze the data. Results. On the results, we found out that Ximenia americana, various parts of Ximenia americana were used to treat 22 human diseases in Eastern Africa region. This is shown in table 1. You can see we have various diseases like the general, to encourage general health, pain, blood, we have anemia. Digestive issues, we have a number of digestive issues being used where Ximenia americana is used. We have respiratory infections. We have problems in the endocrine system, like diabetes, mellitus, and then we have infections of various kinds, parasitic, protozoal, and even bacterial. Ximenia americana is also used in Eastern Africa region to cure issues like, related to wounds, poisoning, and sexually transmitted diseases. So we found that digestive issues were the leading, with 25% cases of digestive issues ranging from diarrhea, oral thrusts, where Ximenia americana is mainly used, up to 25%. Bacterial and parasitic infections followed with 15%. Then we have sexually transmitted infections, 15% also. Then the others are 5%, as seen in figure 1. The plant parts of Ximenia americana mainly used were stem bark, then roots, leaf, then root and stem bark. Some information reported that some people use root and stem bark. Some use leaf, both leaf and stem bark, and then both leaf and roots. The use of Ximenia americana stem bark, again, is posing it, the fact that the majority of the people are using stem bark is another factor that poses this plant to threats of extinction. Methods of preparation involved mainly decoction, boiling, and infusion, almost the same method. That was the leading method with 71%. Pounding and conversion to powder also followed with 14.29%. Therefore, we realized that stomach problems like diarrhea, where Ximenia americana is mainly used, most of these infections are caused by bacteria and fungi and viruses. Literature information on antibacterial activity of Ximenia americana in East Africa was scanty. However, we have reports from other regions, like West Africa, where Mikai 2009 reported antimicrobial activity against all these strains of bacteria, Pseudomonas aeruginosa, and all those. Also, James et al. Has reported these strains of bacteria, that is 2007. This shows that Ximenia americana has got antibacterial activity, and that informs us why it is being used in traditional medicine to solve problems related to stomach problems. In the study by Geid et al. 2005, he found antifungal activity of Ximenia americana against the Candida albicans. This supports the use of this plant in also solving stomach problems, because some of the stomach problems can also be caused by fungi. Leech infestation tonsils are also caused by bacteria, and this has been supported by studies by Mikai, James, also Ramses, and Geid, all of which come from regions outside East Africa, because literature information from Eastern Africa was very scanty on the activity of Ximenia americana. The study by Gaichu et al. 2017 found antipyretic activity of Ximenia americana, and this explains the use of this plant in curing febrile illness, as reported in Ethiopia by Meshiras 2021. In Kenya, Nyangacha et al. 2016 reported antinociceptive effect of the stem bark of Ximenia americana, and that explains the use of this plant in management of pain, as already mentioned, used in management of abdominal pain and joint pains. Marial from Tanzania, Marial 2021 reported that Semenya americana is used in Tanzania to treat diabetes mellitus. We did not find reported activity of Semenya americana in Tanzania or any other country in Eastern Africa. However, a study by Sheta in India and Abdallah have reported in vitro anti-diabetic activity of Semenya americana. And that indicates that Semenya americana could be having anti-diabetic activity. In the study by Geid of 2005, methanol extracts from leaves of Semenya americana were reported to inhibit the growth of Neisseria gonorrhea. And that could explain why this plant is used to treat gonorrhea and syphilis in Uganda, as reported by Yaziki in 2021. Therefore, this review gives a broad information about the pharmacological activities and uses of Semenya americana. However, there were few experimental studies on clinical research and clinical trials on this particular plant, especially in Eastern Africa region. So therefore, it was not very easy to correlate the traditional uses and pharmacological evaluations of this particular plant in Eastern Africa region. Therefore, there is need to evaluate pharmacological properties of this species, as this may be directly related to the species, to the use in traditional medicine. There is also urgent need for conservation measures of Semenya americana. As already has been reported, we have seen that most of the parts used are the stem bark, followed by the roots. The use of those parts can render a plant extinct. Thank you.
Okay, let's appreciate Charles once more. Let's reserve our questions, so we'll ask them after we have listened from the next two presenters. So the next presenter is Chirchir K. Emmanuel. He'll be talking about application of mixture experiment in optimization of cabbage yield using selected organic manure in Turbo, Uasin Gishu County.
Okay, welcome. My name is Emmanuel, and welcome to this conference with that theme, and particularly our study was concerned about the first theme of the conference, that talks about food security and livelihoods. Our study was an application of mixed experiment in the optimization of cabbage, Brassica oleracea, using selected organic manure, conducted in Turbo, Uasin Gishu County. Cabbage is a type of vegetable that is widely used as both for medicinal purposes and also as food. And this crop can grow in diverse climatic conditions, ranging from either warm or cold, depending on the variety. To plant this crop requires that you have fertilizers. And as a matter of fact, nowadays, there's the use of organic fertilizers will actually boost the yield of this crop. But now, these fertilizers are not generally accepted. So as a matter of fact, we had to do a study to look at the effects of organic fertilizers instead, because that is what actually is generally accepted in the world. And in the recent past, the consumption of the crop has really increased due to the increase in world population. Cabbage farming is a very good, reliable, good business that someone can venture in, and at the end of the day, be able to get profit. Nowadays, we are talking about agribusiness, and as a matter of fact, someone can venture in this horticulture and do the farming for the cabbage, and at the end of the day, fetch good profit. However, there are some decisions that need to be done prior to planting this crop. Such decisions include maybe the size of the land, the variety to grow, market, soil type, terrain, and what have you. But the most important of them all is the market, because there's no need you plant and at the end of the day you lack where to sell your crop. But all the same, the plant actually, the market is almost always ready. And the land where to plant the crop is also very important, because the crop needs a land or a place that you are going to grow it, need a place whereby it is it is well aerated, and yeah, it's well aerated. The soil that is required to support the growth of this plant should have a pH of between 6 to 7.5. That is slightly acidic to neutral. And we have different type of the crop, depending with the variety, the number of days to maturity, texture, and what have you. We have different manures. As the topic said, it is using selected organic manures. So we had to do, to select out of the so many manures that we know, we had to do a selection to choose which to apply in this study, whereby oftentimes when you talk of manure, you're talking about animal droppings or urine. And in this case, we selected 4 types of manures, particularly the chicken manure, the pig manure, the goat, and the used tea leaves. And in this study, we call them as X1, X2, X3, and X4 respectively. Now, the method that we used is a mixture experiment, and that's what was an application of mixture experiment, meaning these science that we have need to be applied in agriculture, so that at the end of the day, we have profit out of it. So mixture and mix is a common term, and it is used widely. And as a matter of fact, when you talk of mixture, it means that you are mixing 2 or more things with an intention of coming up with something at the end of it which will be of interest. So in our case, the final component was assumed to be affected by the variable of interest. So our interest, or the variable interest for our case, was the yield of the crop. And in this study, in this mixed experiment, we had to fix, in each experimental point, you have to fix a particular amount so that you measure based on that particular amount. And in our study, we fixed 48 grams. That is, we had to fix the mixture proportion for the manure to 48 grams per experimental point. The methodology that we used, under that we have first of all the useful equations. So the useful equations that we used are, one, we used the canonical polynomial equation, whereby they can be categorized into 2. That is, we have the linear as indicated by the first equation, and the quadratic as indicated by the second equation. Now you realize in those equations that we do not have the intercept term. So in a mixture experiment, we do not consider the intercept term. It is set at 0. So again, we had to look at the use of Sheaf polynomial. And the Sheaf polynomial, you realize that in the equation 3, it mentions of the intercept term. That is, all right, thank you. So we are saying that you realize that equation 3, equation 4, as when you expand equation 4 and remove the intercept term, at the end of the day, we are going to have that as our model that we are going to have at the end of the day. And the most important thing in the mixture experiment is to fit the slope. Now, fitting the slope in any mixture experiment with more than 4 factors is cumbersome. And so this research considers only 4 components, and one of the components was fixed to a given interval. So in this case, we had to fix the 4th, as you are going to look just ahead of us. So the materials that are needed are those are the necessary for this. And the most important thing is that if you look at the second bolt, that is the certified seed, we had to use a single type of variety, that is Flora F1, which we sourced from Eldoret town. However, also the 4 types of manure that were to be tested, we sourced them from the nearby community, or those farmers that have those animals that do secrete those manure, and we undertook that. But prior to selection of the actual design that we used, we had to do, we had to choose from the 2 main type of mixed experiment design, that is simplex lattice design and central design. We had to choose from all the possible designs from the simplex lattice design based on the prominent criteria. And when you talk of prominent criteria, it means that it's D, standing for determinant, A, average variance, E, eigenvalue, and T, trace value. And in that case, you realize that the one that had the smallest figure, that is of the rank value, was 4-4 design. So 4-4 design, simplex lattice design is what we applied in this research. So that simplex lattice design have got 35 experimental points as indicated there. So those are the experimental points within which we were looking at, and the observed response was in the other end. So this research was conducted at a place called Muruguzi Village. Muruguzi Village is found in Ngenyelel Ward. Tarbo Sub-County was in Gishu, and the place is located approximately 36 kilometers from Eldoret town. The soil type there is loamy, and that place is good for planting majority of the crops. Now, this study, we had the assumption that we are assuming, and the following one assumption. One was that the water amount that were irrigated or rain to each plot was constant. 2, the seedling grew at an equal interval. 3, the site soil type is equal in all aspects. 5, the weather and sunshine were affected the seed or the growth of those plants simultaneously, among others. So the implementation of the research were based on these 6 steps. So that is acquisition. We had to do the acquisition of the manure, and we did prepare the land. Measurement were done by use of weighing machine, and mixing was done by hand. Preparing the seedbed also, we try as much as possible to ensure that the seeds that were grown there had the same, that is the height. And also, we looked at those that were vibrant. We did all that to ensure that everything was in order. Spacing and making holes was also done on the same day, and we ensure that all the routine practices were done in the same and single day. Likewise, likewise with harvesting and recording. So these are the results that we had at the end of the day. So if you look at up there, you realize that we have the coded values, we have the actual variables, and we have the average of the cabbage. Now, what happened when you talk of the average of the cabbage? We are talking about the replicates that we have. So the replicates that we had 16 replicates. From each plot, we planted 16 seedlings. Each that is from each experimental point, or each. So we have, from the results, we have that the box plot, it is indicating that the average weight of the crop was between 0.4 kilograms to 3.2. I mean that it ranges from 0.4 to 3.2. We have that the average of the entire crops that we grew were around 2.1. The upper quartile was 2.5. The lower quartile was 1.6. That is the histogram representing the different types of the crops. We had also to fix the regression model, and by use of R statistical package, and that is what we had at the end of the day, whereby our of essence is the estimate of coefficients. And in the estimate of coefficients, you are looking at those that have the positive sign. And if you look at that, you realize that the pure components were the only one that had effect. So that is actually what I've explained there. And from there, you realize that B2 was greater than B1. Same case to that. So let me kindly allow me just go straight to conclusion because of the interest of time. Conclusion. The conclusion is that the study, we used simplex lattice design And in this design, we had that when you look at in pure component, pig manure was the best. However, in the pure double component, the combination of pig manure and chicken manure were termed as the best. Recommendation was that the plotted model is recommended for cabbage farmers, both small and large scale. 2, the study will be recommended that it will be of great importance to apply mixture experiment in mobilization of cabbage using 5 or more organic manures. 4, the study suggests that research could be done in maximizing the horticultural crops such as kales, spinach, and others. And lastly, the study also suggests that maximization of cabbage yield in different locations such as central regions of Kenya, where crop is highly grown. So that is what we have. Thank you so much, and have a blessed day.
Okay, let's appreciate Chichiri once more. So let's have the last presenter for this evening. Please, we'll live together. Let's give him a company as he presents, and then we'll get questions for the 4 presenters at the same time, and then break for the late lunch. So kindly, stick to the time, 10 minutes strictly.
It's the other one. Ile ndogo.
Representation. Sorry, we have a small technicality. Brenda will present tomorrow because our gadget seems not to be compatible with what we have. We'll see what can be done tomorrow. So let's have some time to interact with the presenters. So Brenda, sorry about that, but tomorrow you'll be the first one. So the first presenter, so that we guide ourselves. No, tomorrow. We'll do it tomorrow. Time has we are already, whatever. And she has agreed. So the first one, we stopped at, I think, Mercy Kavila. The title was Physical Inactivity Among University of Eldoret Staff, Kenya. Anybody with a question and a comment on the presentation? Okay, there are 2 comments, Dr. Rachuonyo and then my good friend, Maxwell. So listen to the 2 questions and then you give us the answers. Mercy, we'll allow you to come here.
Can I just talk?
Yes, I think you're audible enough.
Thank you. Mine is just to guide maybe a student talk. When she's presenting at the teaching and staff, she needs to tell us the percentage of those who are not teaching and the percentage of those who are teaching. So that when she comes to the result, say not teaching staff, more of them. And when she says more of them, you need to know the exact number of that more. And I think the last conclusion, I couldn't relate it. With the presentations very well. I don't know, but we can talk behind the tent. Thank you.
So the last conclusion, according to Daktari, it was not captured from your objectives. So you'll respond to that.
Thank you. My first concern would be, what motivated you to do this research at UOE? Secondly, what did you do with 321 sample size to arrive at your results? And that one is in tandem with what Doc has just mentioned.
Okay.
And then, in connection with that, you gave us a multivariate logistical regression model. So we also would expect your work, then at least in comparison with that. And then lastly, of course, it is in line with the statistical work actually, what you did with 321. Thank you.
Okay, so we'll take those two. Mercy, respond so that we go to the next.
Thank you. The first question on what motivated me to do about UOE is because I was targeting institution of higher learning, and UOE is a very good example of an institution of higher learning. Secondly, for 321, from the sampling technique, I was supposed to go to 385 staff, but as you know, about survey is about consent. So if you go to someone and they don't consent to the study, you cannot force them to participate in it. So only 321 were able to consent to it. Then about the percentage of non-teaching and teaching, it is captured in my final project or the essay, so that I can bring it to the next presentation, which is okay. I didn't get clearly what you talked about, the multivariate regression.
Maybe I think for the interest of time, the two of you can interact during
Thank you.
lunch, late lunch. Yeah. Yes. Can I also ask yes, Prof. Prof, yes.
I think it is itching me. Yeah. I did not listen to the presentation to the end. I tried listening online, but it caught my attention because it is interesting. Physical inactivity. Now, the sample that you took, did you categorize your components? Did you categorize them? Because was it made up of lecturers, students, administrative staff, etc.? Because I think different categories of people could have different reasons for inactivity, and they would also react differently. So did you do that? Thank you.
Respond to that question.
Thank you, Prof. Yes, I categorized my components. I was only focusing on the staff in the university. I didn't include the part-timers. And the staff, I had two categories that I was interested in according to the nature of the work, because I was targeting at the sedentarism that has cropped, or the reduction in occupational PA. So I would targeted on the teaching as one category and the non-teaching as another category. Thank you.
Okay, Prof, are you satisfied for now? Okay, good. Okay, we go to the next presenter. Any question for Okumu Naomi? The title of our topic was Datura stramponium, evaluation of its potential as a biocide for mosquito causing malaria. Any question directed towards that? Yes, Prof, and then Maxwell.
Yeah, Datura is a common weed around.
Yes.
And so if it could be having such benefits, it would be very nice for us to make use of it. But I was disappointed. I think the review is not conclusive. For example, the components of Datura that you showed are the common components of all plants. So I think you should have gone further and be able to tell us, or maybe read papers that would tell us what components are actually active towards malaria parasites or something like that, rather than giving a very general review the way you did. Thank you.
Okay. So again, I'll change my plan. I've been informed the other group still have two more presentation. So maybe we'll entertain the last question. Then we give Brenda a chance to present, and then we'll handle the question. So, Maxwell, your question?
Thank you.
Be straight to the question.
Naomi. Which species of malaria causing would be susceptible to this? That I need at least to come out clearly, especially into your introductory. You assumed that a majority would be knowing. Number two, you talked on the resistance. This morning we had an exposure to molecular approach. I think the review, look at it from the molecular approach and see which are these DNA that this datura component would penetrate in order to break the resistance. And then the third one would be, especially for entomology students who are here, this review has a lot of research gaps, especially in the area of results and implications. Probably it would be good at least to liaise with the norm, because I can see a lot of work.
Okay, I think it's okay. So I think you'll interact with the. Just respond to one, and then because of interest of time, we are going for. I think we'll interact during lunch hour, during lunch session.
I
But just
appreciate the questions, and I would want to report that the actual paper has all the details. Okay. But I tried to prepare for a 15 minutes, 10 minutes presentation. Thank you.
Noted. The rest, I think you'll respond. Chancellor, teach medicinal uses of pharmacological activity of Zemenia americana, a synthetic review. This one was also a review. So that we manage time, I think I'll just read the topic. In case you have a question, you'll interact with the presenter. So can we give, yes?
From the Chancellor,
what we do, I'm seeing time is running out.
just an observation.
You can, Brenda, come and sit. Okay. Thank you. Present again.
This one is a different, it's a different one.
So please just try to rush through, even less than 10 minutes.
Okay.
Marakisha, so yes. With 15 minutes. Because the other guys are going for lunch.
Right. Good afternoon, all, or it's evening. I am Kiptui Brenda Jemutai, and I am going to take you through assessment for growth of improved indigenous chicken, reared on sorghum-based rations, and 3 production systems in Uasin Gishu County. Okay, sorry for that. So this, on the introduction, improved indigenous chicken, they are very adaptable and offer great performance and great profits to the farmers. They also provide socioeconomic benefits to the farmers in many rural communities. And then the major classifications of the production systems are 3. We have the free range, where they scavenge outside mostly. The semi-intensive, where they are partly supplemented and partly scavenged outside, and the intensive, where they are fully housed indoors. They are provided feeds, either homemade or the purchased commercial feeds. Now, sorghum was a major ingredient, and sorghum is it's an energy-dense cereal that can replace maize in chicken rations. This sorghum is 5th produced worldwide and 2nd in Africa after maize. The statement of the problem, many farmers rear improved indigenous chicken, so there's high demand on feeds.
the high demand makes the feed cost to rise there because of competition between humans and the poultry for feed. And then also there are high tannin sorghums that compromise with this chicken performance because of the anti-nutritional factors in them. So this leads to low adoption of sorghum as a feed ingredient in chicken rations. The justification is smallholder chicken production provides livelihoods to farmers in terms of income production. There is family nutrition and women empowerment. And then poultry production also boosts the economic development. It also contributes to the socioeconomic status of the farmers through the sales. And low tannin sorghum, it has a great potential such that it can be used as an alternative to maize in poultry rations. The study objective, the broad objective was to enhance production of improved nutritious chicken using sorghum-based rations among smallholder farmers. And the more specific objectives were 3, to determine the nutrient composition of the sorghum-based rations used. I was also determining the growth performance of the improved nutritious chicken under the 3 production systems and the dietary rations that were provided, and to investigate socioeconomic drivers influencing rearing of improved indigenous chicken by smallholder farmers in the study site. The study site was Wasingishu County for the objectives 2 and 3, and for the analysis of the nutrient composition, it was done at a commercial laboratory. So that's the map of the study area in Wasingishu County, Moiben sub-county. Determination of the nutrient compositions, I had 3, the experimental diets were 3. There was T1, that was the 50% sorghum inclusion level. T2 was 75% sorghum inclusion level, and the T3 was a commercial feed, which was a control for the experiment. So on the right is the table for the formulated diets, the T1 and the T2, with varying levels of sorghum, maize, and soybean. The other ingredients were constant. For the lab analysis of the feed rations, 6 samples, 2 from each treatment, T1, T2, T3, were analyzed for proximate and amino acids, selected amino acids. And then the proximate analysis followed the AOAC 2005. The amino acids followed the AOAC official method 2004. Sorghum that was used was also, for the formulation, was also analyzed for tannin levels. For the growth performance, 154 weeks old improved nutritious chicken of the Rainbow Rooster breed were selected and allotted to the 3 production systems. We had the intensive, semi-intensive, and the free range. RCBD, the randomized complete block design was used with a split plot arrangement of treatments, whereby the whole plots were the production systems and the treatments were the subplots. And then weights of improved nutritious chicken were taken bi-weekly for 12 weeks. So that is, table 2 is the experimental layout with the production systems and the treatments. Farmers were also assessed for the socioeconomic drivers influencing rearing of improved nutritious chicken, whereby you selected 15 farmers purposively, those rearing chicken and also utilizing sorghum as a feed ingredient. The data collected of interest was demographic characteristics. We had poultry information, feeding dynamics, the production costs, and the improved technologies and management practices used by the farmers. Data on nutrient composition and growth were subjected to ANOVA, and later means that differed significantly we used Turkey's test to compare the means that differed significantly. And then for the socioeconomic drivers, descriptive statistics in form of means and percentages were generated. So table 3 highlights the nutrient composition of the diets. We had the proximate and the amino acids. So for diet T1, it exceeded the requirement for crude fat because of the high levels of the soybean, which is an oil seed. Also, diet T2 exceeded that because of the soybean in it. However, crude protein was a bit low because the soybean in diet T2 weighed a bit. It was 17.5 kilos, and in T1 it was 22 kilos. So that is why it affected the variation. And then the T3 was a commercial feed. It didn't meet the nutrient requirements for the protein, crude fiber, and cysteine. But all of them met the nutrient requirements for chicken feed for the energy required. That is what I just explained. But the crude protein levels in this study were a bit lower compared to that reported by Masenya et al., which ranged from 16.2 to 18.7. And also lysine content was a bit lower. So figure 4 shows the growth of improved NIRS chicken under the 3 dietary treatments. And that T1, which was the 50%, recorded a significantly high average weight gain compared to the control and the 75%, which was the diet T2. This concurred with a study with the Saleh et al. 2019, where chicken fed with a 50% low turn-in sorghum had significantly higher body weights than those fed with the control diet. So the second one is the growth of improved genetics chicken under the 3 production systems. Here, the intensive, those that were fully housed and supplemented with feeds fully indoors, had a higher significantly average weight gain compared to the free range and the semi-intensive, which concurred with the reports of a study by Gayas et al. 2020, where intensive system had significantly higher growth. For the socioeconomic variables drivers viewed, farmers that were aged 30 to 39 majorly performed the chicken rearing and some other. Also, feed formulations were formulated, homemade feed formulations were made by many farmers, that are 87%. And more farmers used sorghum compared to maize as inputs, and they did not incur this production cost, the marketing and the labor. The improved technology and management practices majorly adopted were the housing and the improved feeds. Those lists include the record keeping. So, in the conclusion that T1 met the most minimum nutrient requirement standards for the ingredients tested, the intensive production system produced a high average weight compared to those other systems. The socioeconomic drivers assessed influence rearing of improved NIRS chicken. The recommendation is that farmers should adopt the Formula T1 and also the intensive system. They should embrace chicken rearing because it also boosts the socioeconomic status. Thank you so much. Those are some of the references.
Just take a seat. Okay. So thank you, Brenda, for keeping time. We'll take questions for the last few present. I think Brenda has just finished, so you should be having the question. So there is one person who was not asked a question. That was Chirchir, yes. The topic was application of mixture experiment in optimization of cabbage. Then there's the scientific name there, yield using selected organic manure in turbo. So any question, any comments on how Chirchir can improve the work? Any missing link, any gap? Yes. So of course, Professor Chodo, and then we go to Maxwell. Maxwell is the only one who is asking questions from the student side.
Yeah. Thank you. I had to ask Chirchir who he is, and I discovered he is a mathematician. So I actually am tempted to keep my comments because they are agricultural. But if this is a thesis, work towards a thesis, he needs a lot of advice from agriculturalists so that he can moderate his model. Because there are several questions that he needs to answer. For example, why did he fix his mixtures at 48 grams, for example? And then he tried to explain that the maximum number of factors should be 4 or something like that. Otherwise, the function will be maybe too complicated. And then I'm trying to look at the factors that he used, and that is where he really needs to look at it technically. For example, the manures that he used. What is the source of the manure? What is the quality of the manure? Because that determines the performance. Because in the end, he's using yield. So he needs to understand what has caused the yield differences that he has, because that will affect his regression equation largely. The other thing that he said that there were 16 seedlings, and he's calling them replications, meaning one plant is a replicate. I don't know what kind of experimentation that is, but maybe it is mathematics.
No, let him clarify, Prof.
So those are the things that I think anybody reading the paper would question. Thank you.
Hey, let him clarify that one. I think I'm interested in that one. 16 samples replication.
Thank you, Prof. Why I fixed, first of all, let me begin with the first question that you asked about fixing at 48. If you read a paper by Barry, 2006, it gave out the ranges within which these organic fertilizers can be applied to different crops. And it was between 36 to 60 grams. So that's why I had to fix mine at 48 grams per experimental point. Now, why 16? What I meant is in every plot, we planted 16 using that very proportion, say pure mixture, let's say goat manure, 48 grams in every experimental point of 48 grams of a pure mixture, say goat manure, we planted a seedling there. So in that particular plot, we had 16 seeds of cabbage planted. So that's what I meant by replication. I hope my point is clear. Okay, the design that we used is simplex lattice design. That is the experiment that we, the design that we used. Simplex lattice design. And basically that resulted from having compared the simplex lattice design. We have in the simplex lattice design, we have 4-2 simplex lattice design, 4-3 simplex lattice design, 4-4. Yes. Then we have also the centroid simplex design, centroid design. So upon comparing them, based on the what we call the prominent optimality criteria, we saw that the best one was the 4-4. That is what we were using.
Okay.
Yeah.
I think I'll advise you to interact more with the professor.
Thank you.
It could be one of your internal examiners for the thesis. Okay. It's not a okay. Okay, Maxwell, ask a question, and then we go to the last presenter.
Okay, thank you, Chirchir. This is an eye opener. You as a mathematician, you challenged quite a number of us. Thank you so much for this great attempt. However, you just have to improve in the why you chose A, B, C, D, isn't it? Especially for manures were selected based on what? Based on support of growth. So that one you can improve to your work. And then also, that one is in tandem with big manure resulted into the best result. So you have to justify that, isn't it? Otherwise, thank you.
Okay, thank you. But on a serious note, Churchill, get in touch with Professor so that he can advise you. I know you're doing applied maths, so you need people from the other side so that your experiments can be modified to come up with meaningful data. We come now to the last presenter. I'm sure that is still fresh in our mind. Any question? Or you need the title? So the title of the presentation was Growth of Improved Indigenous Chicken Reared on Sorghum-Based Formulation Under the Production Systems in Uasin Gishu County, Kenya. That's the title. Any comment, any question? I don't think so. One was not even. One was talking about aflatoxin. This one is talking about effect of improving chicken on that one. I think there's a big difference. This other one is nutrition. Okay. Okay. Any other thing? Okay. So I think we'll still interact as we. I was waiting for the organizers to tell us the plan for tomorrow. Cyrus? Sorry. So I think maybe we can still interact at some point. I know I cut some people short trying to ask questions. As we wait for the organizers to give us the direction for tomorrow, we could interact briefly before we break for lunch. We'll go together. We'll go together. Maybe we'll have. So, any comment? So, I think it's a nice opportunity. You have seen Churchill has benefited. He has gotten some advice that will go towards improving his work. And that is the purpose of this workshop. You present to your peers, your researcher, then they give you some advice on how the work can be improved. I think as we are waiting for the organizers, I'll just wish to remind us that this is what was said earlier, that tomorrow we'll start early. I think we'll have a tree planting session in the morning, and then I think we plan to finish by lunch hour. So all those who have not presented are kindly advised to make their presentations ready for tomorrow. I don't know how many have been done the other side, but here we have done, I think 9, which is about 85% of what we presented today. Okay. So if they are not coming, I think. Or Rapporteur, you have something to say? Okay. Any comments, any reactions? Yes, Naomi.
I'm just appreciating everybody. I have learned, and it has not been in vain. I'm a better student. Thank you.
Thank you. That's a positive remark. Okay. Professor Ochodo, do you want to grace the occasion? We need to recognize your presence. Can you just... No, you didn't... You just say something. I know you cannot lack 1 or 2 words of wisdom.
Okay, Prof, thank you. I just want to appreciate the presenters. I think they have made a very good attempt, but I'm hoping that the comments that people have given them will enrich whatever they are doing. I am happy that we are embracing systematic literature review, which is very important. And I know also that you can do it even better and faster when you use the softwares that are available for literature review. Of course, AI is there. AI is there to help you, but I think it is important to be careful when you are using these softwares for your literature review. You'll find that there are certain areas where they can also lie. And also it is possible that you might need much more recent information that might not already be in the databases that these softwares are using. Because basically, these softwares depend on databases. And so you'll find that there is some very recent information that are still not in those databases, which you still have to use other methods to find out. But I'm impressed that we are moving with technology, and that's beautiful. Thank you, Chair. Sorry I came late.
It's okay, understand.
I was also doing other things.
Thank you, Professor. Thank you. Thank you. Let's appreciate our professor. It's good when we have our seniors around, at least we get encouraged and we know that we are doing the right thing. I think at this particular point, I would wish, I don't want our guest speaker to say anything. Let him not preempt what he'll say tomorrow, but thank you. We appreciate you for coming and also for being part of this team, Prof. Then I also recognize the presence of Dean School of Science. Thank you for being here with us. At least that was quite encouraging. I hope tomorrow we'll see the other deans, or a representative, Dean School of Agriculture, Dean School of Engineering, and Dean School of Environmental Science and Natural Resource Management. Is there? Thank you. Thank you. We know, but there should be somebody in the office acting. That's what I'm trying to say. So can we have a Prof? Yeah, that's how. Thank you. Thank you. Of course, with the permission. Okay. So can we have a volunteer to pray so that we can go for lunch? As you pray, also pray for tomorrow that everything will go as planned, especially with technology and other things. Today, I think it was quite smooth, and we thank God for that. So volunteer, can I volunteer? Okay, somebody has volunteered herself. And also pray for the victims of flood. I think tomorrow, make it a good prayer. Thank you.
Let us humble ourselves and pray. Father, in the name of Jesus, we called on you when we began, and Lord, we have seen thy hand throughout the day. We thank you and we honor you, mighty King of glory, and we commit before you ourselves as we go home, as we prepare for tomorrow. And we pray, Jehovah God, that you take charge. Take charge of everything concerning us and concerning our purpose in this place. We want to honor your name even as we live, and we appreciate you, Lord, for your provision for us for the day. We thank you, Lord. We honor you, for it is in Jesus' name we pray and believe. Amen.